Hey, check what two, check what two. Hey, check, check what.
Check, check. Hey,
Good morning. Good morning, everyone, and welcome to the fourth and last day of our conference. Hope you are enjoying the last 3 days, and we have another exciting day ahead. I hope you enjoyed the dinner last night. We had a lot of fun with good music and good food. And today we are just here really to introduce the last day and just to give you a few reminders. And I would like to remind you to please vote for the Young Researcher Tournament. So today is really our last chance to vote until 11. The voting QR code, I don't know if you'll be able to put it up, but the QR code appeared when during the presentations of the Young Tournament. And you need to rank the different presentations on 5 criteria. And then the final winner will be announced today at the closing of our conference at 4:30. So we really, really appreciate if you could vote on the presentations and let us know who do you think should be the winner of the Young Tournament this year. Also, we would really appreciate if you could complete the evaluation form of the conference. So this really, really would help us to plan for next year and really understand what how you enjoy the conference, what things you would like to be changed. So we really appreciate if you could do these two things during the course of this morning. And so now we are going to our sixth session of the morning, session on vaccine immunology. I'm sure everybody's waiting for the new advances in that area, and I will let Joyce to introduce the chairs and the session of this morning. Thank you so much, and please, I see that people are still coming in, so please take your seats, and we'll start in a moment. Thank you.
Thank you and good morning. So I'll go ahead and introduce the chairs for session 6 of day 4. So our chairs are Tony Pedra. Dr. Tony is a pediatric infectious disease physician, scientist, and professor in the Department of molecular virology and microbiology and pediatrics, and director of the CLIA Certified Respiratory Virus Diagnostic Laboratory and Pandemic Threat Technology Center at Baylor College of Medicine, Houston, Texas. And our second chair is Ruth Caron. Dr. Caron is a pediatric infectious diseases physician and professor at the Bluebank School of Public Health. Her research interest includes the evaluation of respiratory virus vaccines in adults and children. So welcome our chairs so that you may invite our speakers for today. Thank you.
Good morning. Before we start, I would say to the young investigators, a lesson that we're learning today, and that's to be flexible. Because Ulla Borkholz is not able to come here, but Dr. Ruth Caron has graciously taken her place and will be presenting her slides as well. And Barney Graham, who was going to be the closure, is not here today. So I will not take his place. But my point is, we have to be flexible at times. So with that, I would like to introduce our first speaker, Charles Sande, who will be talking about biomarkers of childhood respiratory disease. Charles is a fellow at the Camry Wellcome Trust Research Program. He works at the Centers for Tropical Medicine and Global Health at the University of Oxford, at Oxford, UK. And he has a special area of expertise, like many of us, in respiratory viruses. And he's driven by understanding better the mechanism of pathology and has developed some very unique tools to understand better respiratory syncytial virus, bacterial pneumonia, and other respiratory diseases and mechanisms behind that. With that, I welcome Charles to come to the podium. There he's at, okay. Shows you I don't have my glasses right now. Well,
Thank you very much, Tony. So thank you to the organizers for inviting me to talk about some of our work. And I was asked to talk about biomarkers of RSV, and I thought I would approach this from an angle of looking at the airway microbiome, just following on from the very excellent presentation from Debbie yesterday. So those are my disclosures. So just to set the scene, this needs no introduction to anybody in this room that RSV is a major cause of severe illness, and it's particularly a problem in lower and middle income countries. As we heard from the session led by Harish, the burden of mortality and death is disproportionately skewed in the direction of lower and middle income countries. However, as we've heard of all the exciting new interventions that are coming on stream, it's very unlikely due to financial constraints that all of them are going to be introduced, at least in the near term, into these low and middle income countries. And therefore, we still have, I think, a pretty long way to go in having these interventions rolled out in these settings. So in our eyes, we think live attenuated vaccines as a means of directly immunizing babies is still quite a feasible way of protecting kids from LMIC settings. At the very least, these programs would be far easier to introduce into existing EPI programs because they target kids directly. And there is far more social acceptance relative to maternal vaccination as people have gotten used over the decades to kids getting vaccinated. So we just think the barriers of implementation of this direct immunization platforms would be a lot easier than maternal vaccination. So a key unknown when you're thinking about vaccinating kids directly with a live attenuated vaccine is what it does to the population of bacteria and viruses that are already resident in the airway. And this being a genetically modified organism might have interactions with the airway that are unpredictable. So one way to gain insight about the way this attenuated viruses might impact the airway is to glean insights from natural infection. So I thought I'd put this up just to highlight some of the fantastic work by Debbie Bogart, who's really led the charge in looking at the airway microbiome. And this is a paper from her group in 2016, where they showed very much aligned with what she showed yesterday. Severe RSV is associated with an increased abundance of certain bacterial groups in the airway, led by Haemophilus, Streptococcus, and it appears to be negatively associated with colonization with things like Corynebacterium. And this is a fantastic piece of work, I think, and it shows us very much that the infection of the airway by RSV is not benign, and therefore creates the rationale for assuming that similar interactions might occur with attenuated vaccines. However, I think one of the slight downsides to the studies that have been there so far is that they have been disproportionately focused on individuals in high-income countries, predominantly in Europe and North America. And as we've heard from talks before, there are significant population-specific differences that might impact not only vaccine immunogenicity, but also how we carry microbes in our airway. There are differences in household structures, patterns of social interactions, environmental factors are massively different between individuals in high and low income countries. And this inevitably would impact the patterns of bacterial carriage and how RSV would interact with those bacteria. So understanding patterns of carriage in a localized context is important. So our research interest, at least insofar as this talk is concerned, would be to replicate some of the work that Debbie and others have done in high-income countries to look at how babies in our setting are colonized by bacteria and understand how RSV then alters those patterns of carriage. And in this context, we wanted to look at the microbiome in its broadest guise to include both the bacteria component of the microbiome as well as the virome, to see what RSV's effect on that whole community is. And of course, then start working out mechanistic insights that underlie whatever changes we observe. So the framework for the studies that I'm going to describe are based on the study by Patrick Munywoki, the Kilifi household cohort study, which many of you will be familiar with, but just in the way of a primer, this study was nestled within our local RSV season, which runs right from the beginning of December to around the end of April, May. So we recruited kids well ahead of the epidemic, and we sampled them for 6 months at an interval of roughly 2 time points for every week. So it was very, very intensive sampling. And Patrick has already shown you a little bit of a flavor of what has achieved the enormity of the study. We were able to collect samples at very close intervals from members of entire households. And we then used this framework to try and answer these questions of microbial dynamics within the airway in the context of RSV infection. So what did we do? We looked at some of the kids, infants who are RSV naive were our primary interest, so kids who had never seen an RSV infection. So we wanted to look at the microbiome, and we used a shotgun metagenomic sequencing approach, which allowed us not only to look at the bacteria, but also at the viruses and the fungi that may or may not be resident in the airway. Concurrent with this, we looked at what respiratory viruses were infecting these kids, RNA-based viruses, and this included RSV and 14 other common respiratory viruses. We also collected symptomatic data at each time point to try and grade the severity of illness. And we also measured soluble immune mediators at different time points to associate the host response with the microbial dynamics on the one hand and viral replication on the other. So I'll dive right into what we found. So this word cloud indicates sort of a snapshot, a bird's eye view of the microbiome from our kids in Kilifi. It looks very, very similar to the descriptions of the microbiome from Debbie's group. The predominant species by far, the Moraxellus, followed by the pneumococcus, Dolosigramulum, Haemophilus. Things that we know from Debbie's work are predominant among kids in the high-income country setting. And when you look at, sorry. So when you cluster these kids together and look at whether there are similarities between groups of children, you can see certain themes coming up. So each this here shows it's a stacked bar of each of the bacterial species that we found in the group. Each color is an individual bacterial or viral species in this case, and each individual bar is a child. And the y-axis is a scale from 0 to 100 showing the relative abundance. And when you cluster these kids on the basis of similarity, you can see certain themes. Certain kids are different groups are predominated by different bacterial species. For instance, the blue cluster on the right there is predominated by the pink color, which is Moraxella cataralis. The one next to it is a mix of Moraxella and the pneumococcus, and so on and so forth. So there is quite a lot of variance in the upper respiratory tract microbiota. So what we wanted to understand is, what are the temporal dynamics of this microbiota? Is it relatively stable in time? Does it shift as kids grow older? And what do respiratory insults, including RSV, do to shift the patterns of courage? So this is an example of just one child. And here I've highlighted 5 members of the microbiome: Moraxella actoralis, Haemophilus influenzae, Streptococcus pneumoniae, Moraxella lincolni, and Paramyxovirus 3. And what you can see there is a pattern that is fairly consistent in the kids that we looked at. So the x-axis are recurrent sampling time points, and the blue line is a nonlinear regression fit, just to help us visualize the temporal changes in this, in the abundance of this bacteria and viruses in the airway. So for Moraxella cataralis, you see this quite strong dominance, and that's shown in the first panel in red. In this particular child, it hovered at around 50% across the time course, and it was pretty consistent across the study period. On the other hand, when you look at Haemophilus and Streptococcus, you see this quite temporal incursions, very short-lived incursions into the airway, a bit of replication, and then they go away. Here is another example of child 2. In this case, Moraxella was present at a slightly lower abundance across, but it was present across the board. And you saw similar patterns for Haemophilus and Streptococcus, where you had these short-lived periods of colonization that came and went with time. In this particular example, in the fourth panel, you have Moraxella lincolni, which appeared to be in balance with Moraxella cataralis and remained predominant throughout the sampling period. In the last panel for that particular child, you see human polyoma virus. And this just to give you a sense that this viruses and bacteria are in constant interplay within the airway. And that's the example of a last child, and it's pretty much the same picture. So using this technology, sorry, this hasn't projected quite correctly. We are not only able to see what's in the airway, we can also tell what those bugs are doing. We can infer microbial functional pathways to understand what it is that they're doing and how viral infections such as RSV might alter the generality of the functional generality of the community of bugs that are living in the airway. So in this case, I'm showing you the top graph is an individual child. What you should see there next to Streptococcus pneumonia and Haemophilus influenza are periods when those two bugs are in abundance in the airway. And those periods of abundance are juxtaposed by an increase in one microbial pathway, in this case, carbohydrate degradation. So during those periods of time, the population of the microbiome upregulated that pathway, and it was very specifically linked to those two bugs. So this, using these tools, we are starting to piece together how the entire microbiome works, how it comes together, and understanding how RSV perturbs those patterns. So being an RSV meeting, what effect does RSV have on the microbiota in our local context, in the Kilifi, Kenya context? So very much like Debbie, we found that certain bacterial species were in higher abundance during times of RSV infection. So when we followed up our kids in time and looked at time points when they were RSV positive, we asked the question, are there certain bacterial or viral components of the microbiome that appear to be elevated during RSV infection in a way that they are not when these kids don't have RSV? And that's what you see there. Every bar that is blue that is on the right indicates elevated coverage of those particular bugs in kids who have RSV. So the top ones were Haemophilus influenza, Streptococcus pneumoniae. But to our surprise, we saw a few viruses crop up in that association. We saw herpes virus 5, which is CMV. We saw toctenovirus and strep mitis were also elevated during RSV infection. And very much in alignment with the observations of Debbie's team from yesterday, we saw Corynebacterium, Dolosigranulum, and Moraxella in our hands was associated with healthy children compared to kids with symptomatic RSV infection. And those associations for Streptococcus and Haemophilus are shown here formally in the statistical test. In kids who are RSV positive, in the gray and the brown box and whisker, in the green box and whisker plots, you have much lower abundance than when kids have RSV positive. And this has been shown, and this data are reassuring in reinforcing the fact that you're seeing that abundance of the pneumococcus and Haemophilus in the context of symptomatic RSV. And this is a nonlinear regression plot just showing the same thing because we collected samples before, during, and after RSV infection. We are able to get a snapshot in time of what the impact of RSV is. So here we have synchronized everything relative to day zero being the first day when an RSV positive sample was identified. And what you can see there is the abundance of Haemophilus is linearly correlated with the time of RSV infection and negatively correlated in the case of Moraxella. So I'll just walk through a series of slides just to highlight that in a little bit more detail in this longitudinal study design. So in this case, this is one individual child who was followed between January and June. We had around 40 samples for this particular child. And the arrows, the inverted arrows in yellow there show time points when you had an incidental RSV infection. And what you can see there is the abundance of Streptococcus linearly, temporally correlates quite strongly with having RSV. Here is another example with Haemophilus showing the same thing. So this is a different child, but you saw the same pattern of dominance of Haemophilus during RSV infection, and on and on. We saw this consistently for many of the kids that we looked at. What was surprising in this study was this link with CMV. So in this case, we see another child. This child was infected, had two repeat infections, one that occurred around March and one occurred just before May. And what we see for this child, in the first episode of the infection, we did not see any CMV in their airway, but just after the second infection, we saw quite an abundance of CMV reads. So we followed this in a little bit more detail in all our kids. And this is a summary of some of what we found in a subset of the children. So in all of these kids, although that, so perhaps I should just explain what this is. So each circle shows the abundance of relative abundance in percentage of CMV reads, each of the open circles. The blue line is a nonlinear regression fitted along that. So it shows you the temporal shift in with time of CMV reads. The vertical red lines indicate the time points when these kids were RSV positive. So, and you can see there is quite an interesting temporal correlation between being RSV positive and having an increased abundance of these CMV reads. And this was consistent. So when we tested this more formally, this is what we found when we grouped our kids on the basis of RSV positivity, whether you are RSV positive or negative. There was a strong statistically significant correlation between just being RSV positive and having higher abundance of CMV. And just to say that this does not account for the delayed effects. As I showed you in the first panel, there is potential evidence of a delayed effect in the sense that you might see CMV coming up a day or two after you've seen an RSV positive sample, but this is just restricted to the RSV positive sample. So what we're trying to work out now is the mechanistic associations, the mechanistic reasons for why you have this correlation between CMV and RSV. And the hypothesis we have is that when RSV gets into the airway, it triggers inflammation, and that inflammation then leads to an upregulated expression for the surface receptor for CMV on epithelial cells. So what's the surface receptor of CMV on epithelial cells? This has been a topic of some controversy in previous years, but thankfully that has finally begun to get resolved. And it seems pretty conclusive that at least on epithelial cells, that receptor is neuropilin-2. For other cell types, there might be different patterns of tropism, but on epithelial cells, neuropilin-2 seems to be the definitive receptor for CMV. And that has been shown quite clearly in this paper by Jason McClellan, who did some structural studies and demonstrated fairly conclusively that neuropilin-2 interacts with the CMV pentamer to facilitate entry of the virus into epithelial cells. So why do we think that neuropilin-2 expression on epithelial cells is upregulated by inflammation? So that is supported by studies that have been done by others in other disease contexts that show that inflammation mediated by soluble mediators is sufficient to drive up the expression of NRP2 on epithelial cells. So in this paper, these investigators looked at the MH7A cell line. It's a fibroblast cell line. And in panel E there, they treated these cell lines with different soluble mediators. So on the left there, the control bar is they added nothing. In the next one, they added TNF. And in the next two, they added a combination of TNF alpha and IL-1 beta. And what you can see there, obviously, is just the addition of these soluble mediators in the system is sufficient to upregulate the expression of the CMV receptor. And in panel G and F, you can see very strong evidence that this expression actually results in the translation of proteins. Just having those two mediators is sufficient to upregulate neuropilin 2. So we went back to our samples and looked to see what the expression of these mediators were like in the airway samples that we had. So the panel on the left there is the same child that I'd shown you, but in this case, the red dots and the nonlinear curve represents the expression of IL-1 beta in the respiratory secretions. And what you can see there is that this IL-1 beta response corresponds very much to the peak of RSV replication and appears to decline at the end of that replication cycle. And that was true for the soluble mediators. In this case, I'm just showing you 3 TNF alpha IL-6, but we looked at a panel of 21 others, and for many of them, you saw this pattern. Therefore, just connecting the dots, therefore, if you have this upregulated expression of these inflammatory cytokines in the airway, we think what that's doing is that it's upregulating the expression of NRP2 in epithelial cells and making it more likely that CMV, whether it's through reactivation or whether it's through infection, is more likely to infect those cells. So this, just to push this point a little bit further, we looked at a single cell data set from the literature to see what the correlation was between CMV expression in different cell phenotypes in the nasopharynx and inflammation. So in the top set of graphs, in the top row, is a correlation between NRP2 and, in this case, an interferon-stimulated gene IFI44L. NRP2 is on the y-axis and IFI44L is on the x-axis, and we are using that as a marker of nasal inflammation. And in those different cell types that you see there, we look to see whether there is a correlation. And for 2 individual cell types, ciliated differentiated cells and secretory cells, we saw quite a strong correlation, meaning that if these cells are actively responding to interferon stimulation in an inflammatory environment, they are much more likely to be expressing NRP2. And the bottom set of panels is exactly the same thing, but in this case, we are looking at an irrelevant gene correlating NRP2 with beta-actin expression, and the correlation is lost when you look at that. We also looked again at the lung. So we got a single cell data set from gene expression omnibus and looked to see whether the correlation between the inflammatory marker IFI44L and NRP2 was significantly correlated. And even in this lung-specific airway cells, we saw a strong correlation. So we're still working on this. We're trying, as we speak, we are working to see whether these associations are correlated with airway expression of these proteins in children with RSV infection. And hopefully in subsequent meetings, I'll be able to give you an update. So just in summary, at least in these preliminary studies, there seems to be the hint of a signal that RSV is associated with CMV. The mechanisms, how that works is obviously not super clear at this moment, but we are hoping with this longitudinal studies, we get a little bit better handle on how that association is happening. And we obviously think then that the mechanistic underpinnings of that association is the increased expression of NRP2 in the context of inflammation that's triggered by RSV. And based on the analysis that I've shown you for the single cell data sets, it looks like this mechanism might not be RSV specific. It might be spread across any virus that triggers significant inflammation in the airway. And the implications for this are obviously huge. If this association is right, then it means that vaccines against RSV might have an indirect effect against CMV. So I'd like to thank everybody who's been involved and continues to be involved in the work, colleagues from the Kemri Wellcome Trust, the Oxford Vaccine Group, and the Wellcome Center for Human Genetics, who did all the sequencing work. Thank you.
If this session now is open for questions, please walk. Please.
Testing, thanks. Great presentation, thank you. Just a question about the methods. You mentioned that you use metagenomic testing, but then you also used targeted PCR, RT-PCR for 14 viral pathogens. What sort of concordance did you see there?
Right, so thank you, great question. So the viruses that we targeted with the multiplex PCR were based on an RNA genome. And the method that we used to sequence the microbiome in this case was very much skewed to pathogens with the DNA genome. So we were not able, with our method, with our metagenomic screen, to cover both DNA and RNA-based genomes. And that was the rationale for using separate screening tools for the respiratory viruses.
Louis?
Thanks. That was a great talk. I have a remark and a question. Conceptually, I find it very difficult to tease out these associations, to what extent they are causal. In the first year of life, there is so much going on at the mucosal level that you always find associations. And I'm really unsure how a respiratory virus, by upregulation of a receptor, would really explain the epidemiology of CMV. But your remark is noted that it is something to think about, because CMV is probably one of the most frequent infections systemically, and RSV respiratory. So I thought it was interesting. I would be interested in gut microbiome, etc. My question, and you may comment on my remark, my question is related to the role of the microbiome in vaccine responses. There are very few experts like yourself in the field of respiratory microbiome, so I take the opportunity to ask you here. Immunotherapy for oncological patients has been shown to be really strongly dominated or influenced by the microbiome. Most of these studies are actually gut microbiome more than respiratory microbiome. We are now in the era that we are going to apply vaccines, both live attenuated vaccines as well as subunit vaccines. How do you think the respiratory microbiome will affect vaccine responses, both mucosally applied live attenuated vaccines as well as systemically injected protein-based vaccines? Just your thoughts and what we should do practically from tomorrow onwards to understand the role of the respiratory microbiome. Charles, thank you.
Great, thank you, Louis. So just in response to the first comment, yes, it's very difficult to tease out associations. It's even harder to do it when you have a single time point sample. It's a little bit easier when you have longitudinal sampling and you can see what comes before what. Yeah, so it's a little easier to say what might be triggering what and the direction of that association. So what we're doing now really is to try and piece that together. So there is the evidence from the cell lines that you've seen. Those are very simple experiments that can show you in a very simple mechanistic way, increase in cytokines correlates with increase in receptor. Therefore, you can make that logical connection. Whether that is actually what's happening in the airway is a completely different question. And we're trying to piece that together by looking at that using flow cytometry from children with respiratory infections. With regard to your second question, what do I think the microbiome's impact will be to vaccinations? So I found Debbie's insights yesterday quite intriguing, where she suggested that colonization with things like Dolosi granulum are more associated with homeostasis and allow for a more vibrant inflammatory response that is associated with more rapid clearance of RSV. And I think that alone really puts into context the importance of these things, and that it's probably not wise to ignore such a major component of where the virus is actually going to have its final activity. So I think My answer really is colored by Debbie's observations. I think based on how inflammatory a particular community of the microbiome is, it will possibly impact the infectivity of the live attenuated virus in this case, and the extent to which it can stimulate protective immune responses.
I'm going to ask for one more question in the interest of time. We're actually,
I know, but
well, okay, it's Raj and Kalpit. Stay there then. Go ahead. I'm sorry.
Thanks for a great presentation. I had two questions. The one you've partially answered, but just if you could say it more explicitly, you focused on RSV associations, but then at the end you alluded to the fact that you think that that other respiratory viruses might cause a similar type of pattern. I mean, have you actually looked at that for influenza or other viruses? And then related to that, you spoke about with these viruses causing an inflammatory pathway which sets things off, but we know that sometimes these viruses may be present, perhaps less so for flu and RSV, but something like rhinovirus, which does cause disease, it may cause illness, it may just be sitting there and not cause illness. And in bacteria, we tend to think very distinctly about colonization versus disease. We don't think about viruses that way, but these putative pathways that you're proposing, have you tried to look at whether they occur in the presence of illness? In other words, these underlying inflammatory dynamics are only occurring when the children are ill, or even asymptomatic infections. Do you think these dynamics are happening in the airway anyway?
Yes, so the first question, we were specifically powered just by the fact that the study was designed around the RSV epidemic, specifically powered for RSV. We've looked at human metapneumo, and there seems to be a similar association there, not nearly as strong, but I think the main problem there just comes down to power. The question as to whether these associations are linked to disease, the virus might be there symptomatically, and that is true for RSV as well. You might have, most of the kids do not end up with severe disease. I think the main thing here is not necessarily the individual virus, but the immune response it triggers. If it just so happens to trigger a particularly fulminant response that's characterized by a lot of secretion of these inflammatory mediators, that's the salient point, not the individual virus itself.
Great talk. Raj Selvarangan, Children's Mercy, Kansas City. Two quick questions. One, do you see this association across the lifespan, the viral bacterial interactions? And the second one is it time now with the data that you have and the information in the literature, could the bacterial targets be a good biomarker for, diagnostics as to help with prognosis of illness?
Yes, so the first question, I have to plead ignorance on that one. I haven't seen any data from adults that shows that association, but if anybody in the audience knows a little bit more about this, we'd welcome the intervention. And whether this bacterial associations can act as biomarkers of severe disease, that's a little bit more questionable, because this things like Streptococcus and Haemophilus, we are commensally colonized by them all the time. Therefore, the mere fact that you have an increased abundance in correlation with the virus does not necessarily infer a greater likelihood of invasiveness. So I think that that's a little bit more dubious.
A lot of substantial amount of CMV infection happens congenitally. So have you looked at the relationship of what happens for a CMV positive baby versus a CMV negative baby in terms of their microbiome, including the predisposition to RSV infections?
So we haven't looked neonatally. So these kids were all, in effect, recruited shortly after birth. So none of them appears to have very clear evidence of congenital infection, in the sense that they all looked reasonably seronegative for the first few time points, and that was touched off by the RSV. What's the second part of your question? Sorry.
I think there is a substantial burden of congenital CMV infection. So there are babies born who are CMV positive,
Yeah.
and there are babies who are CMV negative. I was wondering if the reverse is true, that if you have CMV infection earlier on through through the uterus, then you might be more predisposed to some of the other viral infections. I'm talking about a reverse relationship.
Yeah, it's an intriguing question. Haven't looked at that, but it's something to think about. Great, thank you.
Thank you very much. Ruth Karen will now present in place of Ula, but she would like to comment about Ula.
Yes. Hi, everybody. I hope that by now you know I'm not Ula. But I work very closely with her. Ula leads the RNA viruses section of the Laboratory of Infectious Diseases at the NIH. She started working on RSV in 1996 in Germany, and then joined the LID in 2002 and worked with Peter Collins for many years on RSV and human metapneumovirus. She is actually going to give the a recorded presentation. I will do my best to answer questions afterwards. Thank you.
Good morning. Thank you, Sophie, for allowing me to send my talk electronically while I am stuck at home waiting for my first bout of COVID to pass. So I'm going to talk about live intranasal vaccines for active immunization of young children against RSV. These are my disclosures. I don't have to introduce RSV as a pathogen. I just want to note here that a huge burden of RSV illness occurs in children over 6 months of age. There's now 2 passive approaches available for young infants that received approval in 23, including Abrisvo for maternal immunization and long half-life monoclonal antibody nirsevimab. However, both of these approaches provide passive protection that will wane within months, and a pediatric vaccine for active immunization of older infants and children is needed and will probably become part of the pediatric. We are developing live vaccines for intranasal immunization to directly stimulate respiratory immunity, secretory antibodies, local T cells. These vaccines are able to induce rapid protection early in life, and they're immunogenic in presence of maternal antibodies or passive immune prophylaxis, we hope. They are adjuvant-free, and a single dose is substantially immunogenic. And as you all know, killed and subunit vaccines remain contraindicated for RSV-naive recipients because they're associated with markers for ERG. The difficulty with live attenuated vaccines is that they have to be attenuated while maintaining immunogenicity. And in our case, with reverse genetics, we use several ways of decouple attenuation and immunogenicity. Just a couple of slides. We are using reverse genetics to rationally design vaccine candidates with highly defined attenuating mutations, including deletions of non-essential viral genes that oftentimes, in the case of M22, for example, have beneficial effects to upregulate antigen expression, increasing immunogenicity. And we use reverse genetics to combine these elements also to, in some cases, increase antigen expression, F and G expression. So the lead candidate NS2 delta 1313 I1314L is attenuated by deletion of the NS2 gene interferon antagonist and pathogenicity factor, and by a deletion of codon 1313 of the polymerase ORF. This provides temperature sensitivity with a shutoff temperature of 38 degrees Celsius, and the second site mutation preventing compensatory mutations. This candidate was sequentially evaluated in RSV seropositive children and seronegative children, and I'm going to present just quickly the summary of the data from 20 vaccinees immunized by 10 to the 6 PFU intranasally. We saw good safety, no excess respiratory illness in vaccinees compared to placebo recipients. Infectivity, 90% of vaccinees had vaccine shedding detectable in nasal washes with low and moderate shedding titers. The isolates that we were able to obtain were genetically stable, and immunogenicity was very good, with about 80% of vaccinees developing a fourfold or greater rise in RSV serum-neutralizing antibodies. That's comparable to, the titers were low, but comparable to wild-type RSV primary infection. Overall, we concluded that 100% of vaccinees had vaccine tech, and in the following RSV season, we noticed substantial rises in RSV neutralizing serum antibodies during the season in AP. A second line of candidate is attenuated by M22 deletion of the second open reading frame of the M2 gene, which is associated with reduced genome replication and increased viral transcription, in consequence protein synthesis, and we hope immunogenicity. You can see here HEP2 cells infected with delta M22 or wild-type RSV, how much more F and G protein is produced in cell culture in delta M22 infected cells over time. In historic studies, these M22 deletion viruses were highly restricted in chimpanzees, and the gene deletion is, of course, predicted to be genetically stable. In initial studies, we evaluated RSV candidates with M22 deletion alone and found that vaccine shedding was relatively high following intranasal immunization in RSV seronegative infants. Here, let's look at the blue graphs. Then we added additional elements, in this case, the 1030S mutation, another point mutation in the polymerase ORF, which is further attenuating and also confers temperature sensitivity. And we found that, as you can see here in the green graph, this addition reduces the peak level of shedding, and also the whole pattern of shedding is a little bit delayed, and that's another marker of attenuation. So overall lower level of shedding, and of course temperature sensitivity is associated with increased safety in the lowers. Comparing vaccine shedding by immunoplaque assay versus TACMAN assay of the candidates delta NS2 and delta M22, the lead candidates, we found that per infectious unit, delta M22 produces about 10 times more mRNA copies, which would conform with the theory of more mRNA transcription and more immunogenicity. So overall, RSV candidates with M22 deletion generate higher RNA levels in vivo. Looking at the serum antibody response in NS2 data, so the 13 study in RSV seronegative children, or the LID delta M22 1030S candidate, looking at pre and post-vaccine serum antibodies, we found that both candidates induce moderate rises of RSV serum-neutralizing antibodies. This is about comparable to what we would see after the primary RSV infection, wild-type RSV infection. And in this study, about comparable 80 to 85% of vaccinees with a fourfold or greater rise. We also follow these vaccinees over the RSV season with a pre-season and a post-season blood draw to document antibody rises during the surveillance season. And we are in regular contact to capture medically attended acute respiratory illness, which is then resulting in a clinical assessment and virological assessment for to discover if it's RSV associated. Here from the LID delta M22 1030S study, the serial serology pre-vaccine, day 56 post-vaccine, pre and post-RSV season, pre-surveillance and post-RSV surveillance. And we see on day 56 these moderate serum antibody titers that are comparable to those detected in placebo recipients post-RSV season after these kids experienced the first RSV wild-type infection in community. We also regularly find these strong rises in vaccinees during the RSV season, usually not associated with RSV medically attended illness. In this case, we had one case here with the asterisk with an RSV illness. So these are anamnestic responses to wild-type RSV. In this one case, the symptomatic vaccine recipient, interestingly, was the one that was back to baseline before the RSV season in its serum antibody titer. So the second RSV infection in life, we frequently see that we get these substantial RSV serum antibody titers. These are associated with even more solid protection. And we're happy with the primary responses to vaccine, but overall the big question is, can we do anything in addition to get children to this high level of serum antibody titers? So just to put side by side the results from the first RSV Delta NS2 study and the first Delta M221030S study, we find good vaccine take in the majority of participants, low to moderate vaccine shedding by culture and PCR, neutralizing antibody titers, systemic antibody titers similar to those of primary RSV infection, and priming for substantial rises. Vaccine isolates were shown to be genetically stable in both cases, and there's good safety in both cases. The big question, of course, is will these vaccines have efficacy against RSV, MARI, or MALRI? So we looked in a post hoc analysis and grouped many vaccine studies that we had done in recent years to the 5 most promising vaccine candidates, which were those that induced a 4-fold or greater serum antibody rise in 80% or more of participants versus the second group that did not read. This slide shows the pooled efficacy estimates from this post hoc analysis. Vaccine efficacy against RSV MARI is shown at the top graph and against MARI at the bottom graphs. This is combined data from 7 phase 1 studies, and in blue we're showing the combined data from the 5 most promising candidates. The line at the top shows the efficacy estimates for all 160 vaccinees, 47 against MARI and 51% against MARI. When we restrict this to the subset of vaccinees that had a NOT response on day 56, efficacy against MARI goes up to 66% and 100% against MARI. And looking at the efficacy restricted to the most promising vaccine candidates, we found 67% efficacy against MARI and 88% against MARI. And of note, no child with the 4-fold or greater serum antibody response had an RSV MARI detectable. From the study, we also concluded that the 4-fold rise in PRNT is a correlate for vaccine priming and hopefully a good predictor of efficacy. We also extended the post hoc analysis to compare RSV serum antibodies on day 56 after immunization versus antibodies after the following RSV surveillance season, grouping vaccine recipients in those that had post-immunization serum immune responses and post-surveillance further rises, and saw these strong anamnestic responses in the group with putative RSV encounters during the RSV season. In the groups without 4-fold rises during the RSV season, we saw that over the RSV season, the serum antibody levels remain relatively stable over time. And when we compare all vaccinees with 4-fold rises post-vaccine, to all placebo recipients post-surveillance that had an indication of an RSV primary infection, we found that overall primary RSV responses post-vaccine are in the same order of magnitude as those after primary RSV infection. So big question, as I mentioned before, this is what we want to see for real solid protection for the remainder of against severe RSV disease. Since it's relatively hard to boost RSV serum antibodies and immunity with a live attenuated vaccine for non-RSV naive individuals, we're working on vectored vaccine candidates, in this case bovine human parainfluenza virus type 3 as a vector, expressing RSV dsCAF prefusion stabilized either as a soluble protein or anchored in the vector particle. In non-human primate studies, we find that this vector is not inhibited by pre-existing immunity to RSV. And this approach is also effective in presence of passive antibodies to RSV and effective as boost when delivered as a second dose after a primary immunization with RSV vaccine. And this serves as a dual immunization against RSV and parainfluenza virus type 3 pediatric pathogen and respiratory pathogen on its own right. The disadvantage in this approach is only that it's only expressing, this vector is only expressing the single RSV protein, the F protein. In non-human primates, we found in a prime boost study, in this case I'm showing data from 6-month boost, but we've shown this also for shorter and longer boosts, that unlike RSV, live attenuated RSV, shown here at the bottom at the level of detection in nasopharyngeal swabs and tracheal lavages, upon the boost of RSV immune animals, you see no replication, substantial replication of the live attenuated RSV. However, the vectors replicate in African green monkeys to high levels and are not inhibited by pre-existing RSV immunity. These vectors induce a stronger boost, serum anti-RSV F IgA and nasosecretory F IgA. Also serum neutralizing antibodies. In this case, we're showing neutralizing antibody titers without complements. So this is really showing you high quality neutralizing antibody induction of about 8 to 12-fold stronger serum antibody responses post-boost by the RSV-F expressing vector compared to an RSV-F, RSV live attenuated boost. And similar results we also found at boosting at 2 months or later at 15 months. So how do we see live attenuated RSV PIV vectors fit into the big picture in the pediatric schedule? Once passive immunoprophylaxis will wane, we would hope that RSV prime would be provided by live attenuated RSV with all viral antigens, and then possibly followed by a heterologous boost, could also be combined with another RSV boost 2 months later, delivering definitely the pre-F and possibly additional antigens. PIV vectors also would be available in case the passive antibodies will end up interfering with live attenuated RSV vaccine candidates. So we currently have great promising candidates available that are well tolerated, primed for strong memory responses, and show preliminary post-hoc evidence of efficacy post-hoc analyses. We currently evaluate lead candidates in larger phase 1-2 studies to expand safety databases. In addition, Delta NS2, Delta 1313, IF 1314L goes into phase 3 in the hands of our strong creator partner, Sanofi. We are also further characterizing live attenuated pediatric VHPIV vectored vaccines expressing ds-Cath, stabilized and prefusion confirmation. And we'll evaluate phase 1 heterologous prime boost approaches with these vectors. This has been a huge collaboration between different groups from the NIH and our clinical collaborators from Johns Hopkins with Dr. Ruth Karen, Impact, Colleen Cunningham, Elizabeth McFarlane, and others, and our creator partner, Sanofi, with Scott Gallihan and his fantastic team. Thank you very much, and I'm wishing you all the best and a great meeting in Mumbai. I wish I could be there and answer questions.
Ruth has graciously welcomed questions on this excellent presentation. So if you'll have any questions, please come to the microphone.
Hi. Yeah, thank you very much. That was a very interesting talk, Ula, and also great work. I think one of the things I find very interesting about these live attenuated vaccines is that they are applied intranasally. And you have a very good serum antibody response, but I was wondering if you could elaborate a little bit about more local mucosal antibody response and the duration, if you have any data on that, or otherwise hypothesize on the more local immune response.
Yeah. So that's a great question. Thank you. I would say, and I know our collaborators at Sanofi are here and thinking a lot about this question as well. So we would love to be able to look at mucosal antibody. It's challenging in young children. We've, on our end, we've tried a number of approaches using SAM strips, for example, which are my pediatric nurses tell me are, although they're tolerated by hospitalized children, they're not very well tolerated by healthy, normal, active kids. There may be other approaches. I would say in general, I think what is probably true of mucosal immunity is that it's relatively short-lived. And certainly over time, when we've evaluated some of these vaccines, we do see that after 4 months or so, we're often able to boost, which to me is indicative of less mucosal antibody. I think you heard the presentation a day or so ago from Buki, that within a 2-month range, there is a lot less viral replication given with the second dose of the current live attenuated candidate, which to me is a surrogate for mucosal immunity. So I think there is some local immunity, at least at the 2-month mark, but it would be nice to be able to look additionally. Thanks. Cheryl.
Thanks. You've half answered my question, but it wasn't clear from the presentation whether the expectation is that these vaccines are going to block infection, completely, or whether it's about attenuation. In other words, a lot of them seem to be getting boosted anyway. So are you thinking the mechanism is really kids are still getting infected and boosted, but it's not progressing to illness or a mix, or we don't know?
So I am generally not a believer in sterilizing immunity. I mean, it works for a few pathogens. I challenge everyone in this room to tell me one respiratory virus for which we can induce sterilizing immunity. And I think, to your point, Cheryl, those memory responses, those anamnestic responses are indeed indicative of infection under cover of partial immunity that prevents severe disease but allows for boosting of antibody. So, yes, please.
Hi, it was very interesting to see about still seeing boosting for those with pre-existing immunity. I was wondering if it's thought that would still apply for, say, those getting catch-up monoclonal antibodies, say, at 4 or 6 months of age, and if the immunization happens kind of shortly after that, when they may have kind of max more antibodies in their system, or if that's only been looked at for those that it's largely waned.
Yeah. And colleagues at Sanofi may want to comment on this as well. I will just say that as somebody just mentioned earlier, these vaccines are all delivered intranasally. The gradient, at least in cotton rats, the gradient from serum to nose is about 300 to 1. So, so antibody doesn't transudate very well into the upper nasal mucosa. And I can say that clearly it was with less attenuated RSV vaccines than the one that is moving forward in phase 3 trials. But in some of those, in our early studies, we went down to as young as 2 months of age in kids who all had maternally derived antibody, not from vaccination, but just naturally acquired. And what we found in those cases is that actually replication in the nose was not at all blunted compared to older infants who didn't have antibody. It was difficult to detect, say, neutralizing antibody responses because they were under cover of maternal antibody. But in those instances, we did not see it. Clearly, it's something that needs to be evaluated in the current context, though, I agree. Thanks.
Any other question?
Just one comment, Ruth, because with the study that we presented, we looked at the immune response in those had the disease before, so those who were positive. And we saw that the immune response 28 days after vaccination, 2 doses, was similar in those who did not have any response, those who were naive, and those who were positive. So that tells you that there is some response as well. The increase was not that high, but the response was, the GMDs were similar. So that's something to look at. And we'll be looking at the immune response, the mucosal immune response as well. So, thank you.
I will ask Ruth a question. Looking into the future. So it is clear that vaccines and monoclonal antibodies are being developed to protect infants and young toddlers during the first 2 years of life. But we know through good surveillance that RSV is still a significant pathogen through up to 5 years of age, at least globally. What would you foresee additional vaccines for those older children?
I think that's an interesting question, and it has, it's both what is the medical need and what is the programmatic feasibility. I think, and what is the priority? I think all of those are questions. I mean, my hope is that, to me, it's very encouraging to see those memory responses in young children. And one thing that this is a public service announcement, but one thing that I just wanted to say to the group is you have to not, we are used to looking at passive immunity that is entirely antibody derived. So we're really used to looking at incredibly high antibody titers, whether it's through nirsevimab or Brisvo in young babies. We don't actually see, when we look at a primary immune response to these live attenuated vaccines, we don't see antibody levels that are anywhere near that. We see modest responses. But what we do see is that there's evidence of priming. And so when these children encounter natural infection, as Ula said, often they don't come to medical attention, but their neutralizing antibody titers skyrocket. So it's really important to take on board the difference in mechanism for these methods of protection. My hope is that priming may be sufficient to carry these children through early childhood, but I think we won't know until we do the studies and we follow them.
Well, thank you all very much. This session here is over at this time, a little bit earlier. So I would say you got 10 minutes additional to see posters.
Thanks.
I want to wish you that. What? I'm looking forward to yeah, you're gonna be great. I heard that like Melanie like, oh well she wants to I'm like, okay, I need to make sure I tell her good luck. So like have a great day.
Yeah, I think that's okay. They were going to see if you already now. Yeah, playing.
Am I supposed to come up here now or just sit in the front row?
As you wish. I think everyone's coming up. So
I saw Andy up here. Why is Andy up there? What would you like? That's fine. Mine's not going to have enough time left anyway, so.
Yeah, hi everyone again, and welcome back to our next session, which is on lower middle income country health economics and policy. So I'm going to introduce our session chairs, who are Padmini and Neil Rev. So Padmini is the Deputy Director, Bill and Melinda Gates Foundation, Seattle, USA. And Neil is from University Medical Center, Utrecht, the Netherlands. Welcome.
Thanks, Joyce, and welcome everybody to this session. As Joyce said, I'm Padmini Shrikantaya. I lead the RSV strategy at the Gates Foundation in Seattle, where our focus is on supporting the development and then enabling the access to affordable and efficacious RSV prevention products for low and middle income countries. And I'll turn it over to my co-chair, Nele.
Yes, thank you, Padmini. My name is Nele Rave. I'm a third-year PhD student at the University Medical Center, and I have a background in biomedical sciences, and I'm currently working on several health economic studies. In collaboration with PATH, we are performing a cost of illness study and a cost effectiveness analysis in 4 low and lower middle income countries. And it's my pleasure to co-share this session with Padmini, and I will hand over the word to you, Padmini.
Great, thanks. So we've heard a lot over the last couple of days of some great science and great progress in product development for RSV. And of course, I think I won't be the first one to say this is a particularly exciting moment for RSV because we have both licensed and efficacious products for prevention of disease, particularly in the youngest infant population. But in order to make impact, it is not just the licensure of products that we need, it's actually access and decisions and immunization that will ultimately lead to the impact that we are all working towards to reduce disease and mortality, particularly in the most vulnerable. And so to that end, this session is really going to focus on the types of processes and decisions and evidence to policy decisions that need to be made in low middle income countries to enable the introduction of vaccines and monoclonals and ultimately lead to the impact that we want to see. So our first speaker is Dr. Danny Feiken from WHO. Danny is a medical doctor trained in internal medicine. He has a long history of having worked at US CDC, mostly focused on respiratory illness, and including work in Western Kenya for 6 years. And also was one of the leads of the PERCH study, looking at etiologies of hospitalized pneumonia in low middle income countries. He currently works in the Department of Immunizations, Vaccines, and Biologicals at WHO, where he focuses on RSV as well as COVID. And Danny is going to speak to us about moving the agenda forward for RSV prevention from the WHO perspective. So over to you, Danny.
Thank you, Padmini. Let me just make sure I can, okay. Good afternoon, everyone. I'll be speaking about the perspective of the World Health Organization on moving the agenda on RSV prevention forward. I want to start by outlining 5 challenges that I see to the introduction of RSV immunization to protect infants in lower and middle income countries, or LMICs. Challenge number 1 is lack of awareness. RSV does not have the cachet of malaria or even pneumococcal pneumonia. It's not been tested for routinely. It has a clinical syndrome that resembles other pathogen syndromes. And so without this awareness, there's not going to be demand. This is a non-representative survey from Gavi, in which 36 Gavi eligible countries were asked how they would prioritize the vaccines that are currently in the Gavi portfolio or ones that are soon to be potentially available. The blue and the orange bars represent highly likely or possibly likely to introduce. You can see where RSV is in the red box. It's in the lower third right now. So clearly this is our starting point, and we need to do work to move RSV up that ladder. The second challenge is that there's been a historic gap between the introduction of new vaccines between high-income countries and low and middle-income countries. On the left, you can see what happened with pneumococcal conjugate vaccine. The green line represents high-income countries. And you can see within a few years, coverage had passed 50%, and by about 5 or 6 years was over 80% of high-income countries had introduced. In contrast, the red line is for middle-income countries, and the blue line is for Gavi-eligible low-income countries. Over a decade after the first introduction, coverage was still less than 50% of countries. On the right side, for human papillomavirus, it's perhaps even more stark. After 13 years, 79% of high-income countries had introduced, but yet only 21% of low-income countries had introduced HPV vaccines. So RSV vaccines, which will also have a dual market, high-income and low-income countries, faces the same risk of having a gap in introduction. Challenge 3 is that the RSV immunization products don't fit neatly into the existing childhood immunization schedule. On the left side here, you can see the timing of vaccines to protect children. For pregnant women in these countries, it's really only tetanus toxoid-containing vaccines that are widely used. Although influenza and COVID vaccines are recommended, they're not used very often. At the newborn point of contact, when RSV monoclonals would be given, it's only BCG and more recently hepatitis B birth dose that are given. In contrast, the infant immunization schedule, which in many countries is still 6, 10, and 14 weeks, there's up to a dozen vaccines that are given. These were the time points where the pneumococcal conjugate vaccine, rotavirus, Hib, could be plunked down right on top of the existing schedule. Fourth challenge. There are competing immunization priorities in these countries. This is a crowded neighborhood. And although in this room we may all think RSV prevention is super important and exciting, it's not necessarily the case. Countries in Afro are very excited about malaria vaccines. They're trying to increase coverage of inactivated polio vaccines, measles second dose, HPV. And during the pandemic, there's been backsliding in routine immunization, and so there's a big catch-up on routine immunization. RSV immunization has to find its way into this crowded neighborhood. And the last challenge is that the policy pathway is more protracted in LMICs. There are extra steps, as you can see on the bottom in light blue. This is where WHO plays a role, and you'll hear a talk later today about from Gavi about addressing the financing step. So with these 5 challenges in mind, I want to talk about what we're doing at WHO to address some of these challenges, starting with lack of awareness and competing priorities. Well, one of the things we hear most often when we've talked to policymakers and regional counterparts is we don't have any disease burden, RSV disease burden from our area, so we can't make decisions. Well, I think we all in this room would say, of course you have RSV. RSV is everywhere, but many countries need to see data. So to address that effort, there's several surveillance initiatives that WHO is involved in. The main one is expanding the GISRIS, or the influenza surveillance platform that's been in existence for decades. Over the last 6 years or so, RSV surveillance in children has been introduced into 25 GISRIS countries and will continue to be so going forward. In addition, we're trying another approach, which is piggybacking onto vaccine preventable disease surveillance that's been ongoing for pneumococcal vaccine and rotavirus monitoring. And we hope to start that at Sentinel hospitals later this year in Vellore and one site in West Africa. This might be another approach to fairly quickly get surveillance data. The other approach is that there actually is lots of data out there, even though people don't recognize it. This is an example. This is from the global mortality burden analysis that was done a few years back. Each red dot represents a country at that time that had information on RSV ALRI hospitalization, over 100 sites in every WHO region. And so we've asked Leo Lee, who was the principal investigator on this, to try to repackage this information according to WHO region. And this is some of the data that he's produced. So on the left, you can see rates of RSV-associated ALRI in the community. So of course, it's in every WHO region, as you can see, and it's the highest in the Afro region. In contrast, if you look at the middle graph, what it shows when you look at RSV-associated ALRI hospitalization, it's the lowest in Afro, the blue, which is the blue line. So it's no surprise, high in the community, low in the hospital, that the RSV mortality would be highest in the Afro region, as you see on the right-hand graph with the blue line. And this is the type of existing data that we hope can be repackaged and delivered to policymakers in the regions. One of the mechanisms I discussed yesterday is the RSV Roadshow. I'm not going to discuss it again. It's a package of materials, slide sets, scripts that can be delivered. There's plenty of information outside you can find about the Roadshow. So the next challenge is that these RSV immunization products don't fit neatly into the existing EPI schedule. Well, fortunately for the maternal vaccine, having the maternal vaccine coincides with efforts from the maternal child health community at WHO to improve the quality and quantity of antenatal care visits. In 2016, the guidance went from 4 visits to 8 visits, as you can see on the far right in the table. And at least half of those 8 visits are in the window when you would give an RSV vaccine. And over 100 countries have now adopted these new 8-visit guidelines. As part of the same effort, there's an initiative to increase early ultrasound in countries before 24 weeks. Now, this is done to assess high-risk pregnancies, but it also can allow better establishment of gestational age, which will be useful later in pregnancy for vaccination, as well as post-authorization surveillance to look at preterm births. Along with this, we have worked some with PATH to develop a protocol to assess gestational age of women coming into clinics. This is based on earlier work that was done in Kenya. The idea is that this would get the numbers of women coming in the right window to be vaccinated, so a country could get a sense of what to expect in terms of numbers of women to vaccinate. And this can be used by countries along with other tools like the checklist you heard about yesterday to prepare for introduction. I want to talk about the policy pathway. So an important step along that pathway, and one that is in WHO's remit, is to make a WHO policy decision or a vaccine position paper about a vaccine. And the way this is done is through SAGE, which is the Strategic Advisory Group of Experts on Immunization. SAGE is the principal advisory group to WHO on vaccination. They deliberate, they make a recommendation that becomes the vaccine position paper of WHO. Importantly, a SAGE recommendation is a required step for Gavi to fund or UNICEF to supply vaccines. Now, SAGE has already heard about RSV twice. One was back in 2016 when we were waiting for the Novavax maternal vaccine. We know that story. More recently, in October of 2022, SAGE heard about the newer products, the Pfizer maternal vaccine and RSViNET. And SAGE recognized the huge global burden of RSV and the impact that preventing it could have in LMICs. However, they did also note that there were many competing health priorities in countries and that the pivotal trials did not have sufficient LMIC representation for countries to recognize themselves in those results. So they recommended that in parallel with the regulatory and the policy process, that there be an impact study that would clearly demonstrate the full public health impact of these vaccines or immunization products in countries in terms of child health and health systems. And their second recommendation was that we go ahead and form a SAGE working group. Now, what is a SAGE working group? This is a smaller group of people who have more RSV expertise or subject matter expertise who can really dive deeply into the evidence, including benefit risk analysis, grading of the evidence, and come up with a recommendation to the full SAGE about what to do. We formed this group. They've started to meet. We have a lot of work to do this year, and our plan is in the fall of this year to present the data for a recommendation by SAGE on the RSV maternal vaccine, because that will be the first one available to LMICs. Now, the impact study that was also recommended by SAGE, work is also moving in that direction. This will be funded generously by the Gates Foundation. We're working with them to select 3 or 4 sites, mostly in Africa. This will be a real-world trial, so there'll be few exclusion criteria. It will be randomized, and it will focus on the severe outcomes, so RSV-associated and, importantly, all-cause severe LRTI and hospitalization. These are the outcomes that will move policymakers. It will also evaluate preterm birth and low birth weight for the maternal vaccine. I should emphasize this is for the maternal vaccine. Hopefully we'll start later this year in South Africa, and we at WHO will have a role going forward in translating this impact study results to the policymakers. I want to talk about pre-qualification. This is a process that started in 1987, has been done for over 150 vaccines. Many LMICs will rely on pre-qualification to authorize a vaccine in their own country. This is more like a regulatory process that WHO runs separately. And again, importantly, UNICEF won't buy and Gavi won't fund any vaccines that have not been pre-qualified. So this is a process that goes hand in hand with the SAGE recommendation. As I said, it's a regulatory step. However, if there is a stringent regulatory authority that has already licensed that product, which is the case for the RSV products, this process can be as quick as 90 days. There is an extra step, however, and that's something called programmatic suitability for pre-qualification. And that's because vaccinating kids in these countries is more challenging. There's constraints that are not the case in high-income countries. So I want to highlight the example of an auto-disable syringe. Because there was so much bloodborne transmission from reuse of needles, part of pre-qualification requires a auto-disable syringe, and the current products are not in auto-disable syringe formulations. For the maternal vaccine, Pfizer is developing a single-dose vial that can be used with an auto-disable syringe, and they also have partnered with the Gates Foundation to develop a multi-dose vial, which will ultimately be the preferred product for these countries. And lastly, I don't want to overlook the regional technical advisory groups on immunization and the national advisory groups, the RITAGs and the NITAGs. You heard about this some yesterday, and as Rose Gelongo put it, this is really where the rubber meets the road, because this is where the decisions get made about which vaccines a country will use. So we really need to make sure we reach out to these groups and that we can assist them in their policy processes. So in summary, right, the time for the beep, there are significant challenges to overcome. We, and not just we, but many of us in this room are working to overcome these challenges, improving awareness of RSV, overcoming the programmatic obstacles, and accelerating the policy process. And we hope that within the next couple of years, starting with the maternal vaccine, followed hopefully soon after by a monoclonal, the first LMICs will introduce RSV preventive products. Thank you.
Thanks, Danny. The way we've envisioned this session is we'll have all 3 speakers present their talks and then have a panel discussion afterwards so that you can raise all of your questions at that time. So I'll turn over to Naila to introduce our next speaker.
Yes, thank you. So now it's time for Andrew Clark, and I'm very honored to introduce him. I sometimes call him the man of the UNIVAC model. And yeah, Andy, you can already come to the front, please. Andrew Clark is Associate Professor in the Health Decision Modeling based at the London School of Hygiene and Tropical Medicine. His research basically focused on the development of analytic models to inform decision about new vaccines. Most of his work is in low and lower middle income countries and involves supporting country-led evaluations of the potential impact. He has been working on cost effectiveness for rotavirus, human papillomavirus, and RSV. And the model ranged from simple Excel-based decision support models through the more complex individual-based models. And these models have already been used in more than 30 different countries. And today he will introduce us to the UNIVAC model that can be used for RSV prevention and intervention strategies when performing a cost effectiveness analysis. Thank you, Andy.
Okay, thank you, Nile. Thank you, ReSViNET, for inviting me to talk. This presentation is all about cost effectiveness of the new long-acting MAB and the new maternal vaccine in low and middle income countries. I have no conflicts of interest. So why consider cost effectiveness in the first place? I think when most people think of a health economist, they think of someone who's a bit miserable, unfeeling, putting a price on life. But actually, most of the health economists I know are pretty compassionate, and they're interested in maximizing the number of healthy years of life you can gain for a fixed budget. So essentially, they're trying to get good value for the fixed budget that's available. So somewhere on the right-hand side of this slide, is where a health economist wants to be. And you can argue for more health budget, but eventually there will be a ceiling and you have to work within that ceiling. So how can we determine if RSV interventions are good value? Most cost effectiveness analyses will end up on a graph that looks like this. So you've got costs on the y-axis and health benefit on the x-axis. Health benefit is DALYs averted or QALYs gained, but essentially it's the number of healthy years of life that you're gaining in the population. An RSV intervention may have a certain cost and health benefit, but the crucial thing is the ratio, so the cost, how much it costs to gain every one healthy year of life. That's the primary outcome measure of the cost effectiveness analysis. And you can compare that ratio to different RSV strategies, different interventions, other vaccines, other health interventions. Alternatively, you can compare it to a willingness to pay threshold. So really you want to get that RSV intervention into that zone of what you're willing to pay. And not many countries have a formal willingness to pay threshold. We've got one in the UK. The countries that do have one, it tends to be around 1 GDP per capita or less. So it just gives you a rough idea of the middle income, high income countries where they tend to set their thresholds. If you want to get the RSV intervention into that zone, the options are to reduce the dose price or maybe be a bit more creative with the strategies you use, maybe a seasonal or targeted strategy. So you need a model to do this, a vaccine impact and cost effectiveness model. This is one of the models that are out there. There's lots of different RSV models out there at the moment. And it's actually really good practice to use different models and see what differences they give you when you run your analysis. This one's called UNIVAC. It's called the Universal Vaccine Decision Support Model. And it's been, I guess what makes it different is that it's been specifically designed for use by national teams in low and middle income countries. So it's completely built in Excel to be accessible. It's translated into different languages. And it's designed to be projected onto a screen like this in a stakeholder workshop so that different stakeholders can agree what the different inputs should be, giving the country more ownership over the analysis so they can defend their numbers. The process is that the NITAG or its equivalent would set the timeline for appraising the RSV options, that establish an RSV working group, work on the model and the evidence, and then present that back to the NITAG. And that process has worked quite successfully with UNIVAC in quite a few countries with rotavirus and HPV vaccines. For RSV, we've done 2 completed studies now, Argentina and Vietnam, and I'll show you some results from those studies. In addition, the RSV Gold Health Economics group are working in Nigeria, Cameroon, Mozambique, and Nepal using UNIVAC to look at the cost effectiveness of long-acting MAB and maternal vaccine. It's quite a simple model. It's deliberately been designed to be simple. It's a cohort model that calculates results with and without the different RSV intervention in each week of age from birth to age 5 years. It has no transmission dynamics. It's really important to be clear about that. We do develop models with transmission in them, but for this purpose, I think you heard on Wednesday, there's still quite a bit of uncertainty about the role of transmission. So we've decided to focus on the direct effects of the MAB and the maternal vaccine. Births are split into groups that have the intervention and don't have the intervention, those that have severe disease, non-severe disease, those that have clinic visits, hospital admissions and deaths, and so on. And that data comes from things like RSV surveillance in the country, ALRI incidence rates, the systematic review we heard about a couple of days ago, and other sources. But the important thing is those sources are agreed by national stakeholders, so they could defend those numbers. In terms of cost of illness, this is something that can really strengthen a national study. The RSV Gold Network are doing cost of illness studies in Ghana, Nigeria, Mozambique, and Nepal. And Argentina and Vietnam have also collected cost of illness data on RSV. Another important input to the model is the age distribution of RSV for different presentations by week of age. And on the right, you can see a couple of examples of some fits to the data we have. I think the top one's cases, the bottom one, hospital admissions. And we're in the process of trying to gather as much data as we can on RSV age distributions in low and middle income countries. And that's coming through something called the RSV Age Study Consortium, which is WHO, London School, Edinburgh, and Nanjing. There was a systematic review, and WHO have now sent letters out requesting investigators to see if they're willing to share their RSV data. So if you've already responded and shared your data, this is my opportunity to say a big thank you. If you're still deliberating whether to share, please talk to myself, Danny, or Harish, and we can tell you a bit more about that study. Another big input is the real-world timeliness of dose administration. I'm showing you DTP1, DTP3, and measles here, but the one I'm really interested in for MAB is BCG, so the green age distribution at the front. I think a lot of people think BCG is given bang on time at birth, but actually the average tends to be about 3 weeks, 4 weeks, sometimes up to 10 weeks. So it's important to take that into account when you're modeling the impact of the MAB. And that data comes from DHS and MIX, and we've got data for about 80 countries. Efficacy and waning, we're using the data from the clinical trials. In the base case, we assume a fixed efficacy for 6 months for the maternal vaccine and 5 for the long-acting MAB. We also run what-if scenarios to see what would happen if we used a more gradual decline in efficacy over time. So in terms of results, these are results for Vietnam. So this is a year-round maternal vaccine strategy. And we've got 3 price points here. So you've got $25 a dose at the top, $15 a dose, and then $5 a dose. The pink dot is the point estimate, and the cloud is your probabilistic uncertainty. And then you've got 3 lines that represent possible willingness to pay thresholds. As a group at the University of York for Vietnam, they would recommend about half a GDP per capita as their threshold. And on that basis, $15 a dose would be too high. Maybe something closer to $10 would work in Vietnam if you use that particular threshold. If we add the infant MAB, you can see the picture gets quite complicated. There's quite a lot of overlap in the uncertainty clouds. And actually, it's very hard to differentiate cost effectiveness between the 2 products if you assume the same dose price. We have greater impact and greater cost just because we assume greater coverage for MAB. For Argentina, Same thing, a year round. This is quite new data that was shared by Mauricio's team. A year round maternal vaccine strategy, this time with 3 price points at $100 a dose, $50 and $25 a dose. And in Argentina, they do have quite a respected economics group that have set a threshold at near 0.9 times the GDP per capita. And on that basis, a maternal vaccine at $50 a dose should be an acceptable price for a maternal vaccine in Argentina. Again, if we add the infant MAB, it's a confusing picture, similar cost effectiveness if you assume the same price. Again, slightly greater impact and cost. In Argentina, they do have good data on the date of birth and the date of hospital admission. So we looked at how much the cost effectiveness would improve if you didn't do a year-round strategy, just did the first 6 months of the year. So this is showing impact if you incrementally add a month for each month of the year. And you can see the impact tails off after about June. You're not really getting any extra impact after that. If you add another axis showing dose efficiency, you can see it's much more cost effective. There's kind of this sweet spot of the first 6 months of the year where it's very cost effective and you're getting pretty similar impact to the year-round strategy. There's a lot of caveats with this, but we wanted to do this to sort of show potential difference in dose efficiency. This is sort of getting away from country-specific analyses. We also did a more crude desk analysis of all low and middle income countries. And this is showing the percentage of low and middle income countries that might be willing to pay for a maternal vaccine if it was priced at $3.50 a dose. And you can see that about 30% of countries, we found that it would actually be cost saving. So it would be the right thing to do to introduce it. It'd actually save money. For the other countries, assuming a willingness to pay up to half a GDP per capita, they would all be willing to pay on that basis. The dotted line is a separate analysis that was done by Path using a different model, and it just shows pretty consistent results between the two models. If you increase it to $7 a dose, then you still have quite a few countries with cost-saving results. But on the right-hand side, there are about 10% of countries where they wouldn't be willing to pay, assuming a willingness to pay of half a GDP per capita. So in conclusion, decision support models provide a framework for country teams to build consensus on data inputs and strengthen the local ownership of their results. And the main uncertainties really are all around dose price and the feasibility of these seasonal and targeted strategies. They aren't going to work so well if there's lots of variation in seasonal patterns across the country, if it costs a lot to implement that particular strategy. Thank you for listening.
Yes, thank you, Andy, for this great talk. We will keep the questions until the end of the session, and I will hand over to Padmini to introduce our third speaker.
Great. Well, it's my great pleasure then to introduce Cheryl Cohen, who is a professor in epidemiology at the University of Witwatersrand and also heads the Center for Respiratory Disease and Meningitis at the National Institute of Communicable Diseases in South Africa. She leads national surveillance and generation of evidence to guide policy for control of respiratory diseases and meningitis. And today we'll be presenting to us now, if we had heard about the global level from Danny and on cost effectiveness and cost effectiveness analyses from Andy, we'll get a specific country perspective from South Africa from Cheryl. So over to you, Cheryl.
Good morning, Andy. Thanks for the introduction and thanks for the opportunity to speak at this meeting. These are my disclosures. And just to start off with, I think everyone's familiar with this data. And in fact, this is the same message that Padmini started the session with. We know there's a lot of work to show the massive burden of RSV in terms of acute lower respiratory tract infection and mortality globally. But the overwhelming majority of these cases are concentrated in low and middle income countries. And if we are to impact on these severe outcomes, we have to ensure rapid access to low and middle income settings. This is a study that was published last year It's a case control study from 4 low and middle income countries, really in a range of settings, different continents. And in the study, they enrolled hospitalized children with acute lower respiratory tract infection and healthy controls in the community. And what you can see overwhelmingly from these 4 sites, and this is the picture really wherever you look in the world, is that RSV overwhelmingly dominates as the main contributor to the syndrome of acute lower respiratory tract infection. And not only is it the top cause, but also it stands out in relation to all of the other causes. We conducted a similar study in South Africa, and in this study we enrolled young infants aged less than 1 who are hospitalized with lower respiratory tract infection, and we enrolled healthy controls. We also tested for many respiratory viruses, although I'm just showing a few pathogens on the slide. What was different in this study was that we didn't only enroll children hospitalized with respiratory illness, but we also enrolled children hospitalized with a medical admission, but there was no respiratory diagnosis at all. And the reason we did this was we were interested to understand or quantify the contribution of the pathogens typically associated with respiratory presentations to this non-respiratory burden after we adjust for prevalence in healthy controls. You can see here the results of our study. Firstly, we confirmed a very high attributable fraction for RSV, and again, RSV standing out way beyond the other contenders as the top cause of acute lower respiratory tract infection. But interestingly, we also found a significant attributable fraction for RSV amongst non-respiratory cases. It was the only so-called respiratory pathogen that was statistically significant. And after adjusting for this attributable fraction, we estimate approximately 9% of non-respiratory admissions are also attributable to RSV, presumably because RSV is setting off a cascade of events which result in illness or perhaps presenting non-specifically with febrile seizures or other presentations. But I think this adds to the different strands of evidence which suggests that introducing these vaccines in low and middle income settings may well have positive impacts way beyond the simple proportion that we detect on our PCRs. This is another attributable fraction study from South Africa published last year. It differs from the data I've just shown you in that we're not only focusing on children less than 1, but in this study we're really looking across the life course. So we've enrolled children or individuals hospitalized at any age. And also we're not only looking at severe disease, your right-hand graph, the SARI is hospitalized illness, but we also looked at outpatient disease, the influenza-like illness. Again, we enrolled healthy controls, and we tried to determine the attributable fraction across the life course. If you focus on the dark green line, and for this talk, just focus on the severe disease, you see very beautifully that U-shaped distribution of attributable fraction with the highest attributable fraction in young infants and then increasing again among the elderly. And also what you see in the graph, if you look at the background shading and you see the darkest green, the little wedge on the left-hand side, you see again, the very high contribution of RSV in this study, over 40% of admissions among children less than 1 attributable, but also just to draw your attention to the fact that that wedge really goes all the way to 5 years of age. So while the highest burden is really in the youngest children, there is also a substantial burden in children up to the age of 5 years. And I'm just going to talk a little, I've got just 3 slides on this, and then the rest of my talk will really be about the interventions in infants, but I wanted to just quickly, we discussed it this morning, so I just wanted to talk a little bit about older kids, because I think as people have mentioned with this morning's talk, when we start thinking about vaccinating children over the age of 6 months, we can consider that there are not only going to be benefits preventing disease in that group, the direct benefits, but there could also be indirect benefits in terms of reductions of transmission within the population. And these type of indirect effects for other vaccines like pneumococcus, of course, had massive impacts in terms of cost effectiveness and benefit. So we conducted a study in South Africa prior to COVID. It was called FIRST. It was a community cohort study in an urban and a rural site. And in each site, we enrolled 50 households each year and enrolled everyone in the house. And we took nasopharyngeal swabs from all household members twice a week and tested them on PCR. So a very intensive community cohort study to understand burden and transmission in communities, really modeled on Patrick Muniwoki's study in Kenya, which you've heard quite a lot about here. I thought this audience would be interested. The data have actually been published, the RSV data were published in January of this year. But what isn't published, or at least directly presented in the paper, is a comparison of the two important respiratory viruses, influenza and RSV. And I think it's quite interesting to look at the differences and the similarities between these two viruses in terms of the key transmission parameters that you pull out with these intensive studies in a setting such as ours. I think the first striking result is the very high burden of infection, over 40 per 100 person years in both sites for both these pathogens. RSV a little bit higher, though, in terms of infection incidence. When you sample people so frequently, to our surprise, we find a relatively high percent of repeat infections. 10% of RSV, of individuals with RSV have a second episode in the same year, usually with the other strains, so RSVA versus B, and they're usually very young. Symptomatic fraction for RSV, 1 in 3 episodes are symptomatic in our setting, and a relatively low proportion of those are medically attended. And interestingly, the transmission parameters are almost identical for these two pathogens. So on average, if you have 1 infected case in the household, 1 in 10 susceptible household contacts will get infected. And symptomatic people much more likely to transmit, but asymptomatics also transmitting, and because asymptomatic infections are so common, a very important contributor. Shedding duration are almost identical between the two viruses. And if we look now across the age spectrum, so on the left you have flu, on the right RSV. The top graph is incidence, and the bottom graph is a symptomatic fraction, percentage of episodes with symptomatic disease. The blue colors are infection episodes. Dark blue is a first infection, light blue is a repeat infection, and then the green shades are different flavors of symptomatic disease, and the red shades different flavors of medically attended illness. The top graph of incidence, you see how although RSV disease, severe disease, really concentrated in the extremes of age, you really are seeing infections right across the life course. Although, of course, by far the highest incidence in young children. But these infections in older children, and the peak incidence is in fact 1 to 4 year olds, are of course driving the transmission in the community. When you look at the bottom graph, though, of the proportion of cases that are symptomatic, so your gray is asymptomatic, you see this very strong, particularly for RSV, U-shaped distribution, with the propensity to develop symptoms given infection being greatest in the extremes of age. And this is really the converse of that attributable fraction graph I showed you before. And so what we've done, and this is an influenza graph, hopefully next meeting I'll be able to show you for RSV. We've developed a transmission model that's been parameterized on the data from our burden and transmission study to explore the potential effects of vaccinating young children with a 50% coverage of a 50% efficacious vaccine against transmission to explore what would be the potential impact of such a vaccine. And what we did is we compared our findings for South Africa to a model parameterized with data from the United Kingdom. And this is early data, it's work in progress, but it does suggest that in our setting, indirect effects could be substantially greater. The percentage reduction in unvaccinated people could be substantially greater, really driven by our age distribution and contact patterns in our setting. So I think in the future, really potential for impact in low and middle income settings. But now I'm going to move to the products that are to hand. So this is maternal vaccination and long-acting monoclonals. And we've heard through this meeting already some of the early data on the dramatic impacts that are being seen in high-income countries from these interventions. So I thought I'd just give you a bit of a flavor of what's going on in South Africa. South Africa is an upper middle income country. We have a temperate climate, and RSV is highly seasonal. Our season is really just starting now. It occurs in the Southern Hemisphere winter. We have a strong health products regulator, the South African Health Products Regulatory Agency, and we pay for our own vaccines. We're not a Gavi eligible country, but we do have a strong history of self-funding costly but important vaccines such as the pneumococcal conjugate and the rotavirus vaccine. We have a strong NITAG called the National Advisory Group on Immunization, and the NITAG advises on new vaccines. And we do strongly consider guidance from WHO. Danny spoke about SAGE. We take SAGE very seriously, but we certainly have the independence to make recommendations that deviate from stage as well. And our NITEG has already, in fact, constituted an RSV vaccine working group to consider the question of RSV vaccines, because we recognize the extremely high burden. So I thought I would just take you through a little bit of the type of data that a country NITEG would consider or that we are considering in our decision-making process. This slide summarizes data on the burden of RSV in South Africa, and you can see the 3 endpoints: mild disease, severe disease, and mortality. There's a few key things to take away from this graph. I think the first is the extremely high burden of severe disease in our setting. Severe disease estimated at a rate of over 14,000 per 100,000 person years. The second, and also mortality, we estimate about 600 children die of a birth cohort of 1 million each year. The second important point is that is how heavily the data are skewed to the youngest ages in our setting, with really the peak in the first two months of life. And this, of course, is extremely beneficial when you have the short-acting or these interventions where the efficacy reduces with time since administration. And then thirdly, the high proportion of severe cases that are occurring out of the hospital. This is, in fact, much more marked in some other African settings, but even in South Africa, 1 in 3 deaths occur outside the hospital, and about half of all severe illness episodes occur outside the hospital. And of course, these interventions have the potential to increase equity because we have very high coverage, for example, of the birth dose of BCG. And so even children who may not get to health facilities if they are ill could potentially benefit from these interventions. Another important aspect is cost effectiveness. We've just had a talk on cost effectiveness, and we conducted this study in collaboration with partners from the London School of Hygiene and Tropical Medicine and colleagues from KEMRI in Kenya to look at the cost effectiveness of in red, you can see the monoclonal antibody, and in blue, the maternal vaccine in South Africa and Kenya. Just to focus on, in the red box is South Africa. And you can see here that in South Africa, in fact, at the lowest price point, these interventions could even be cost saving in our setting. And this is really driven by the very high daily burden of mortality, as well as to some extent the high cost of hospitalization. But even at the highest price points, these interventions could really potentially be cost effective. And if you look at the Kenya data, it really is a very similar picture. So really pointing to the potential for these products. Andrew showed this slide in his presentation. So this is from Vietnam, also a middle-income country, lower middle-income country, really striking, in fact, the similarities in terms of the cost per daily averted in their setting. And again, the conclusion that depending on the pricing, these interventions could well be cost effective. So I think that there's really a big body of evidence in certainly in middle-income countries and also coming up in lower-income countries that there is an immense potential benefit and quite a lot of evidence to support countries implementing. What is the current status? I started off by saying, many high-income countries are already introducing, and Danny showed the slide in terms of where WHO SAGE is sitting. In the 2022 meeting, they requested, they said there's a need for more data and requested a trial, which is already starting. I think the big concern from our side is that while there may be more data needed, we also already have a lot of data to show the incredibly large burden. And the real priority is that we not delay introduction of these life-saving products to countries with the trial. So whatever data are needed, I think we need to try and generate them as rapidly as possible. In South Africa, we really sit, we have a big package of data already to support introduction. Yesterday, Pfizer presented that there is some concern about the imbalance that was seen with the maternal vaccine in terms of low birth weight increased low birth weight in vaccinated children. And this was particularly seen at the South African site, although not statistically supported, and it may well have been a chance event. So South Africa is considering to what extent we should delay because of that safety signal. And really, just to highlight also that while maternal vaccines are currently being seen as the top priority for low and middle income countries, monoclonal antibodies really should be considered in our setting. You saw Andrew presented, there's not much difference in terms of cost effectiveness, and they offer many benefits. So to conclude, infant RSV prevention could have substantial impact in many LMIC, and the high burden can lead to favorable cost effectiveness scenarios. Pricing is really critical, and monoclonals should also be considered. And really just to acknowledge the funders and collaborators of our first study and of our burden and transmission study. Thanks.
Thanks, Cheryl. I invite you and Andy to join us here for the panel discussion and invite our audience members to come forward with their questions. Go ahead, Jess.
Hi, thanks, Jess Atwell, Pfizer Global Scientific Affairs. A couple questions about the complexity of the models that compare the two interventions. And so one was just to ask, it sounded like you said, Andrew, that different coverage assumptions are made for maternal versus MAB in those analyses comparing the two. And I'm just curious if you can speak to how that influences things. But I think the more important question is, when thinking about potential burden of disease averted, we all know that severe RSV is more common in the youngest infants, and the cost of caring for very sick young infants is also greater than for older infants. For the vaccine, we have efficacy based on age at illness. For the MAB, it's time since dosing, but not age at illness, right? So knowing the efficacy for infants from birth to 1 month of age, for example, hasn't been published yet. So I'm curious how you incorporated that into those models to think about some of those complexities, because it's very difficult, I think, to think through. So I'd love to hear your thoughts on those two pieces. Thanks.
Yeah, good questions. In a simple static cohort model, coverage is essentially scaling up the costs and the benefits up and down. There's no real other reason for having it in there. Where it does make a difference is it can reflect budget impacts implications. If you've got higher coverage, you're assuming, mainly because we're using BCG as a proxy rather than ANC for the maternal vaccine. So yeah, that's basically what's going on with the coverage. The other question, could you repeat that? I didn't quite.
Yeah, absolutely. So we know that severe disease is more common in youngest infants, right, in the first couple of months of life. And we know that the cost of caring for severe illness in younger infants is more expensive than the cost of caring for older infants who are sick, right? For the vaccine, there are data available on vaccine efficacy by age at illness, right? But for the MAB, it's time since dose, right? And there are some big categorical stratifications of they were, 0 to 3 months when dosed and then out to 150 days and 3 to 6 months when dosed and out 150 days. But to my knowledge, there have not yet been data shared publicly on efficacy by age at illness, not age at dosing, but age at illness. And so I'm just curious how that complexity is accounted for in your models or in others, or how we should all be thinking about this, because I think it's one big complexity in trying to directly compare two very promising interventions, right? Just because of how they were studied.
So we have a different age distribution for severe cases versus ICU versus deaths. And in each week of age, we're assessing what the efficacy and the coverage would be in that particular week of age. And we're dropping the efficacy as you progress through the age. For both the MAB and the maternal vaccine, we're essentially assuming maximum protection straight away at birth, and then using the follow-up efficacy data from the trials as a basis for doing the modeling. So it's not really any more complicated than that.
Thanks.
Thank you so much. Very interesting. And so again, regarding the modeling, and for you and for Cheryl, so when you try to model this type of cost effectiveness analysis in countries where we do not really have data on the effectiveness of the intervention, so South Africa with the HIV rate in pregnant women of almost 30%, in Nepal with high malnutrition rates in pregnant women. So how do you model that if you put that in the models? So we know that for other vaccines that HIV is a big contributor for protection. We know that in data from South Africa that HIV-exposed infants have highest rates of burden of disease, and they have for some of the vaccines lower immune response to some of the vaccines. So I'm just wondering, at this stage of the race, how can you use this type of information in your models, if you do use it at all? Thank you.
Yeah, I think the purpose of a decision support model is that you do run different what-if scenarios, and you're seeing how does that shift the dial in terms of cost effectiveness. So you've got your probabilistic cloud around your cost effectiveness, but we also do a separate kind of chart showing what-if scenarios. So what if the efficacy is much lower than we used in the trial? What if it's much higher? What if it lasts longer? What if it declined gradually? So we look at those different things and see, is it important and worth investigating a bit further? So that's the way we deal with it.
So thanks for us. We haven't considered that, so we assumed it was similar, but it is an important consideration, and there is a trial starting, in fact, in South Africa to look at the efficacy in HIV-infected women. So I absolutely agree, and potentially the benefit might be greater because they're a very high-risk group. So once those data are available, I think it's quite relatively straightforward to compartmentalize the model into the different groups, but for now we haven't considered it.
Go ahead, Louis.
Danny, you explained very carefully that we need an impact trial to understand safety and efficacy of the maternal vaccine in lower middle income countries. Immunogenicity may be different, transplacental transfer antibody may be different. We also have a map. We may soon have a second, maybe soon thereafter a third one. What would be the bridging studies required for pre-qualification and use of those long-acting maps? Because I think many of the concerns for an active vaccine may be different for a map.
Mm-hmm. Well, I'll try to address your point. I think for the impact study, I mean, we've thought about which product, and I think the decision was to be practical and real world about the product that would be available. You know, I think when Sage initially made their recommendation, the focus was mostly on demonstrating impact on health outcomes, and the safety part of it came later as more of that data came to light. So I would say that probably the impact data will be translatable, because if you're preventing early RSV disease, be it by a maternal vaccine or a MAB, that impact from a country standpoint could be translated to a monoclonal antibody. Obviously, the safety issues would be different, but I think what we're hoping with the impact study is that the impact on disease would be translatable to a future monoclonal that would be available. Do you want to chime in?
No, I was going to ask you and Cheryl a question, actually, as you've brought out impact from the different endpoints that have been generated in trials, as well as the types of endpoints that you're considering in this impact trial, or the types of endpoints you've been considering in the NITAG in South Africa. What do you think are the most important for LMICs, or what has been sort of most salient in the discussions in the South African NITAG?
I mean, I think that's always the contrast. I mean, for us as scientists, we trust the specific endpoints, right? It's prevention of RSV-associated LRTI. But I think back to pneumococcus, right? The most impactful was impact on all-cause mortality, even though it was a much smaller percent. So I think, we may not be able to demonstrate that for RSV, but all-cause hospitalization, all-cause LRTI, that's the impact, because that's, from a policy perspective, that means you can close your ICUs. That has a real cost benefit in terms of services. And I think the impact will actually be shown, the trial will certainly add, but it's the before-after in a couple of settings where they have robust data from before. And same as with rotavirus, you see, it will be visible to clinicians. That's what we anticipate with RSV. But it's those non-specific endpoints, I think, that policymakers care about.
I would agree with that. I mean, I think we talked a little bit about all-cause outcomes yesterday, and if we can get an extra bonus beyond RSV by secondary bacterial infections, that would be great. But I mean, we know that RSV causes so much of, up to a third or more of respiratory admissions to hospital, and overall admissions. So if we could get rid of those, I think we'd see I think administrators in hospitals would recognize this clear impact. So I would agree, all-cause outcomes are really the most important for policy.
Hi, thank you for the presentations, all the presentations. Andy, I got a question for you. One of the main problems that we have in Argentina, as you know, was establishing the willingness to pay threshold. Which is the best way, the more transparent and technical way to establish that threshold?
Well, that's a difficult question. Yeah, so there's a group at the University of York, Carl Claxton's group, and I think they're sort of leading the way on the way they think it should be done. And they're essentially looking at the current spending of the health service and the marginal benefits you're getting for each sort of marginal spend, and saying that should be your threshold. You don't want to be introducing interventions that displace other interventions that are doing better than the one you're introducing. So that's the method they've used. It does lead to quite low willingness to pay thresholds in some settings. But there are other economists that use different methods, and it's very complicated. So which is one of the challenges, I think, with communicating this stuff. There's a lot of controversy about what those thresholds should be.
Hi, Peter Richmond from Perth. This is a question, I guess, for Andy and Cheryl, perhaps. With maternal immunisation, the mothers will have antibody that will be persisting beyond that pregnancy. And the question is, particularly given the high incidence of disease in that first few months, having residual immunity from a previous pregnancy where you were vaccinated may actually significantly impact on that second child. And I'm just wondering whether you've included that in any of your calculations, and how critical it is for us to be following up these immunised mums to see what are they like 12 months, 24 months, 36 months post-vaccination. And wonder if you have any comments on that.
Yeah, I did this. I think this was mentioned on Wednesday as well. This sort of quick, rapid turnover of births in some settings. Yeah, we don't count for that. I think the DHS, a mix, we could be looking at intervals, but at the moment we're assuming there's no benefit. You'd have to start again.
Yeah,
I don't know, Cheryl.
And I agree. We don't look at it. I think they also are no data. So I think that would be, and it sounds that it was asked on the first day. I think the trials didn't look at it. So I think it's something that people should try and look at in the observational studies post implementation if it's not going to be done as part of the trial, so that we could have something to quantify in our models.
I guess in South Africa you do have a lot of participants in these pivotal trials that could be followed up to do those bloods, perhaps.
So I have a question for you, Cheryl. In your presentation, you talked about a 10% attributable fraction to RSV in non-respiratory admissions. And I wonder if that was during the RSV or respiratory season or throughout, and how does that inform the decisions about seasonal versus year-round administration of vaccination for you?
So thanks. I didn't have time to talk about seasonal and year-round, but so that it was a one-year study. So it was 10% over the whole year. So within the season, it would be a greater contribution. I think in South Africa, the issue of seasonal versus year-round is really important. And in the model that I presented, we considered a year-round vaccination strategy. From other countries, in most settings, seasonal vaccination is more cost-effective. But I think the programmatic impacts in a setting like ours are substantial and really should not be understated. It's not clear to me whether in South Africa it would be possible to achieve a high coverage with a seasonal program that you switch on and you switch off. We try with influenza, and we don't succeed. And so if we think of the vertical nature of our struggling health systems, there are a lot of reasons why an all-round intervention, if you want to achieve high coverage for such an important disease, I think seasonal strategy, cost effectiveness in models is one thing, but whether it can actually be done in settings with weaker health systems, I think really we need to demonstrate that in different countries before we start saying, well, we're actually saving, because in the end it might just you destroy the whole program because people are so confused and you can't do the supply chain. And there are lots of, I think that aspect really needs to be more studied and then tested in a few settings. And we'll have to work out how to study it without putting in support that then when it's not there, means that you can't do it at the end. Thanks.
I was hoping to follow up on the seasonal administration maternal vaccination. With a 24 to 36 week gestational age, we really struggled with, in the United States, of what that might look like given the potential broad range of dates where infants might be born. And only when it became a 32 to 36 week approval did that seem somewhat feasible. So I don't know if you've found reasonable, if when you've been looking into seasonal administration of maternal vaccination for a more broad 24 to 36 weeks gestational age, if you found reasonable possibilities of what that might look like.
So I know, I mean, I've heard the US struggled, and that's why I think if the US struggled, we'll really struggle. But we've done quite a lot of modeling, in fact, for influenza, seasonal maternal influenza vaccination, and you really lose a lot in terms of, because you don't know women's dates, and it's really. I mean, that's another benefit of monoclonal, right? Because you know when you're giving it and you know when the baby's born. So we've not done a lot with RSV, but I do think it's very challenging. Others may have more insights.
The only thing I would say is that I agree with the programmatic considerations. I mean, if South Africa is going to have trouble, and a lot of other LMICs, it's going to be even harder. But the seasonality of RSV, as you move away from temperate zones, becomes more splayed out over the year. There are peaks with seasons, but there tends to be 2 peaks or more constant circulation. So from the epi standpoint, in a way, it makes more sense to have a year-round approach than it does in temperate settings.
So I have a question for both you and Cheryl related to administration, which is, thinking about a gestational age window, whether it's 24 to 36 or a different, how is South Africa, or how, Danny, from a WHO perspective, are you considering gestational age assessment as a prerequisite, or when women are coming in and just using that as sort of your marker of when you anticipate vaccination can take place? And related to that is the issue of, in many countries, tetanus is a recommended maternal vaccine. So considerations around co-administration, which may be straightforward for tetanus. I don't know if there are other vaccines also recommended in South Africa for pregnant women.
I'll start, then he can give a more comprehensive view. I think, Heather's standing there. She can maybe talk more than me about the gestational age assessment, but I think it's very difficult. Gestational age assessment and the quality of it is extremely poor. Most clinics aren't able to do anything particularly robust. And so from a programmatic perspective, again, you need very simple directives, and you also need to get the patient while she's there. So I think it really needs to be simple, and I don't think it's feasible to do assessments regularly. Your other question was around co-administration. I think is a concern for South Africa. So we are in fact introducing maternal pertussis vaccine currently, and we also have an influenza vaccine recommendation. Although I've mentioned it's really not very well done because of the campaign nature of it. So I think again that's an area where there's not a lot of guidance. In fact, and certainly SAGE hasn't said anything, but I think there does need to be, from a burden perspective, RSV is clearly the highest burden. But there does need to be some clear guidance about co-administration, and if not, sequential administration, and how you manage that, because that's also a real complexity, again, for the nurses in antenatal clinics who are not, other than tetanus, they aren't like EPI, they aren't trained in an EPI way to deal with multiple vaccines in a complex way. So it's a big training and programmatic issue, I think.
Yeah, from the gestational age standpoint, I mean, clearly it will be an issue. When there was an age restriction for rotavirus vaccine, that was an issue in many countries, is adhering to that. So I think if there is a strict gestational age window, that will be difficult in many settings. There are not early ultrasounds that are done. There's other methods for dating gestational age. So I think programmatically, the thought is that eventually we would have to, there would have to be some wiggle room there, probably on both ends of a gestational age window.
Go ahead, Heather.
Thanks to all the speakers, really important presentations. This is a question for Danny, and the issue of translation into policy in low middle income countries and the lag traditionally from what has happened in high income countries. And so the question is around, we saw this lag much reduced in COVID. What are the lessons from COVID? What are the processes that happened in COVID that can be brought to bear on policy changes for implementation of these interventions in low middle income countries?
That's a good question. I think we'd need probably sociologists and philosophers. But I think you're right, Heather, that I think there may be a way to capitalize on that, basically that need and the sort of the glaring inequity that was seen with COVID vaccines and the push, because the world saw the dire situation. And I mean, you've been a great voice to sort of raising the alarm for the dire situation with RSV. And if there's a way we could translate that urgency to policymakers, to donors, to companies, I think that's what we need, is that kind of effort.
Yes, with regards to the time, I think there are no questions from the audience. I actually have one question for Andy, more general questions. As we know, and we have seen from Danny and others during the conference, there's a lot of burden data available, but there are also countries where no burden data is available. So how to handle those missing values in the UNIVAC model?
Yeah, it's a good question. I think this kind of fallback is to use the Harish and Leo's systematic review, which is good data. But ideally, there is some surveillance or ALRI incidents that you can use in country. And I sort of go back to the scenario modeling. You're kind of running it with high and low estimates to see how it changes the cost effectiveness.
Thank you. And with that, I'm going to thank our speakers and also my co-chair and all of you for your attention and your great questions. And we won't stand between you and lunch. I hesitate to say we need more feeding, but off we go.
You all, that's the touch of really, really.
Hello, everyone.
Are you ready for the last session? So it's really a pleasure, to introduce the last session of our meeting. And this session, it's called Global Health Challenges in RSV Prevention. And I think we left the best session for the end. And this session will be shared by Naveen Thakur, the president of the International Pediatric Association in the US, and Soborn Bor from the University of SGT here in India. So I'll give the floor now to the chairs to introduce the speakers for this last session. Hope you enjoy. Thank you.
Thank you. Good afternoon. I'm so happy to see so much audience because this is mostly not expected in the last session, but I know this is a very, very dedicated crowd, and I have attended previous conferences, and so it's a wonderful to see you all here. This session is about global health challenges in RSV prevention. Most of the things have been discussed, but still we have a very excellent lineup of speakers, including me, who will still talk which has not been discussed. So I'm happy to introduce my co-chair, Dr. Sobha Brur. She is emeritus professor currently at SGT Medical College, Gurgaon. She was in All India Institute, have teaching experience of 45 years. Her interest is in virology, molecular diagnosis, molecular epidemiology, and she has received lot of awards, which is, if I start, it will be too long. So I hand over to Dr. Sobha.
Thank you, Dr. Naveen Thakkar. It's a pleasure to be here and chair this last session with him. And the first talk will be by Dr. Naveen Thakkar on the overview of global challenges. And as you know, he's president of the International Pediatric Association and also of the India Child Health Association. And he has been, one of the top influencers for the polio eradication from India. So he's going to talk to us about the global challenges in RSV prevention. So over to Dr. Naveen Thakkar.
Thank you, madam. So you have been here in this room for 4 days. You have heard everything about RSV. I want you to come out of this room and see what's happening around the world. I'm not going to talk what are the specific challenges for RSV prevention, which has been discussed a lot during last 4 days. I am going to discuss what's happening in immunization space around the world, and we cannot be just ignore that. So this is overview of my talk. So this was the slide presented by Kate O'Brien in last EDVAC meeting, and I found it so good that it very well summarizes why we vaccinated adults during COVID, but who got left behind? Children. So this was before the data of WUNIC was released in 23, but we have an imperative to act, and we cannot ignore that. So after that data for 22 was released in 23 of WUNIC, and DTP global immunization coverage in 22 recovered to 2000 partially, but not fully. But still, it's still, there is a backsliding on immunization. And measles, which is called canary in the coal mine, which very well exposes, in immunization gaps. And this is the picture what we have. Measles across the world. Very large outbreaks, including in India. And also we are seeing a lot of mumps cases. There are other outbreaks around the world. This is what CVDPV outbreaks. So if we cannot ignore that, We got last case of polio in 2011. Until that time, we just continued with 6 antigen in our universal immunization program because, all the resources were drawn towards polio. And if this measles, polio, cholera, and other outbreaks continue, forget it that we can get any resources for RSV or any other new vaccine. So this cannot be ignored, and this has to be dealt with. So vaccine preventable outbreaks is a crisis among many crises: food, energy, war, economic, climate, natural, humanitarian, and all the resources are being drawn. We already suffered in COVID, and then where the resources are going. It's a huge crisis. Second is climate change. Again, we cannot be just ignore this because this directly affects. There is a lot of talk now about climate change and its effect on the, infectious diseases. We are seeing cholera outbreaks which has not reported for so many years. So floods, famine, and so many other things, heat, too much cold temperature. What we see even in India, a very cold winter, and also very hot summers. And you have already seen what happens in Europe and other parts. So we cannot ignore that. This has to be dealt with. Humanitarian crisis. This is a very serious issue, and this displaced population is particularly vulnerable. And then also there is a violence in conflict situation where the health centers, health workers are targeted. So you can see 10 million infants remain unvaccinated in humanitarian setting, and how many are displaced. We just cannot ignore this. Inequity in immunization, and our speakers will talk about it, but these are mostly remote, rural, urban poor, conflict affected, and then gender barriers. I'm not talking about the girl child in some countries not getting vaccine. I'm talking about everything we need to address this at all level: literacy, decision making, because this is, if we have seen in India, in Kerala, where education level is very high, the children are mostly vaccinated. So this has directly to deal with the immunization, and we have to address that. Health system challenges. The cost already we have seen. As I said, most of the things have been discussed. Data availability and quality, political will, low digital literacy, and other health system challenges. Vital registration, still many parts have paper-based registration. No doubt India is doing very well, and now we are learning from our experience in COVID to a web-based system where every child will be tracked for immunization. Health workers, we have shortage of health workers everywhere. They are essential, they are crucial, but they are poor conditions, they are poor salary, and limited knowledge. If you ask them, forget RSV, for other vaccine, if you ask them what disease this vaccine prevents, many of them, they don't know. Many of them have not seen. I mean, this this current generation has not seen many of the diseases. Like you just heard about me, polio, because I saw in 1 month, I saw 55 cases of wild polio virus in 2018, 90, 91, 92, and that motivated me. 30% mortality. I saw children dying in front of me, and that I got motivated. Yes, 55 cases in 1 month. Every child coming to my OPD, first thought would be, oh God, let it not be a case of polio. So my colleague from WHO has already said that we have no awareness about RSV. So it's a first thing. You talk about vaccine, you talk about intervention, but people first should know. Even very poor awareness in the medical community. Medical professionals are the most trusted voices. And if they don't know, we have learned in polio, they will say anything about it. So first is the awareness and perception. perception of the severity of disease and need for the vaccine and intervention. And then comes the acceptability and access. Infodemic was already there, we know, and now we have more of AI and deep fakes. It is going to be there. We cannot run away from that. We have to work on digital literacy and work more and more on that, because these things are going to happen. There is a very good book I read about stolen focus. Our focus is totally stolen. You are forced to land to the sites where you will be shown what they want. So we have to live with that. Vaccine hesitancy is my favorite topic. I will say only three things. It's a not new phenomena. History of vaccine hesitancy is as old as history of vaccination, 200 years old. Second thing is, arguments have not changed. People have studied. For last 200 years, arguments have not changed. It is same. It means that beliefs have remained remarkably constant over the better part of 2 centuries. And third thing, it is not going away very soon. Once you have started RSV, and whatever might have happened and whatever happened in past, it will immediately, someone will come up, this vaccine is like that, and this, and we always get that. So this is not going to get away soon, and we have to tackle that. Everyone has to work on that. This is again an old slide by Neil Johnson, and what he has shown, that medical professional and say vaccine advocates, they talk amongst themselves, but anti-vax, they talk, they connect. This is from the Facebook, 1 million user. They are connected across the world, and the way they are expanding, this was before the pandemic, so they will take over us if we don't, all of us don't start talking about vaccine and use social media, not only for personal or family, but also for to advocate vaccine. So to summarize, pandemic has created major disruption, particularly in low and middle income countries. Climate change is imposing its effect on global warming. Conflict and humanitarian crisis has affected immunization, and then hard to reach population, gender balance, economic status is a major challenge. Vaccine hesitancy is still a problem and will continue. Manufacturing capacity, poor resilience of health system are some of the major cause of vaccine inequality. All these barriers, including low awareness and data and other things, will definitely affect. And we have to, digitalization of healthcare system, proper surveillance, data management are crucial. Engagement to build political will is crucial to maximize vaccination. We have missed the opportunity. We don't see anybody here from the government. We don't see here anybody, even not many people from the people who advocate vaccine in India. So maybe we have to arrange another meeting in India, and we will invite all of you again to come to India and make a case for RSV. Thanks to all these people who have helped me in this presentation. Thank you so much.
Thank you, Dr. Thakkar. We can take one or two questions now, or we can take all the questions at the end of the session.
Next is so. Next, we have Anuradha Gupta, who is not present here. Anuradha is the president of Global Immunization at Sabin Vaccine Institute, a veteran public health leader. She has, before this, was deputy CEO of Gavi. Before that, she was additional secretary in Government of India. She has done a lot of work in say polio eradication, launching many new vaccine in India, and we have a video from her which we will run.
Good morning, everyone. I'm so delighted to be joining you virtually, and my apologies that I could not come in person to be with you today. And I'm extremely delighted to be presenting about how we have to be more mindful of certain aspects relating to equity while addressing this new undertaking of rolling out and scaling up RSV vaccines as a part of maternal immunization in low and middle income countries. As you are already aware, a lot of children die each year due to RSV, and 95% of these deaths happen, unfortunately, in low and middle income countries. We also know that the United States of America, where RSV vaccine has already been rolled out as a part of maternal immunization, it hasn't really been easy for pregnant women to access the vaccine. There have been all kinds of challenges ranging from a lack of availability of the vaccine, the financial impediments because of high out-of-pocket costs, and also wrong use of product. One thing that is important to keep in mind is that the RSV vaccine is entering the immunizations. Good morning, everyone. I'm so delighted to be joining you virtually, and my apologies that I could not come in person to be with you today. And I'm extremely delighted to be presenting about how we have to be more mindful of certain aspects relating to equity, is that the RSV vaccine is entering the immunizations.
Hello. Can you sort this out? Otherwise, we can go ahead with next speaker as of now. We'll go ahead with the next speaker. Our next speaker is Dr. Andrew Pollard. Sir Andrew is the director of Oxford Vaccine Group at the University of Oxford and honorary consultant pediatrician at Oxford Children's Hospital. He received a knighthood in the Queen's Birthday Honours in 2021 for services to public health, and the Order of Medical Merit from the Federal Republic of Brazil in 2022. We know very well he led the clinical trial of Oxford AstraZeneca COVID-19 vaccine, and I personally thank him because I was also first person in Gujarat to get your vaccine, sir. So please go ahead.
Thank you very much, and thanks to the organisers for inviting me to be here, and also to the chairs for hosting this session. So I've been asked to speak about RSV implementation in low and middle income countries, and I wondered why I was asked to speak about low and middle income countries, since there's plenty of you who live in low and middle income countries. And then I started to think, well, maybe people are looking at the UK in a slightly different way these days. And I looked at the news headlines yesterday, and saw that our GDP has been falling. We're now in recession. So maybe it is appropriate that I speak to you about low and middle income country implementation. So if we look in the UK at RSV across the life course, you can see here that in the early life, there's between 20 and 30,000 hospital admissions per year. And similarly, there's a large number of ICU admissions in young children. But the reason why I know I'm not living in a lower middle income country is that when we look at deaths, they don't figure at all on this chart. And so this is the typical pattern in a high-income country where RSV is a problem for the health system, but doesn't have a high mortality associated with it, as you can see here. And you've already seen several times Harish's work on the global burden of disease. And I think there's two important things to highlight here. First of all is the enormous number of hospital admissions globally, around 3.6 million estimated in this analysis. But perhaps even more staggering is the 100,000 RSV-associated deaths. And as you were hearing from Aniruddha, 97% of those deaths are in lower middle income countries. So this is a pretty fearful situation that we're in at the moment with RSV from a global perspective. Problem for our health systems everywhere, but for severe mortality and morbidity in LMICs. But the good news is that we do now have interventions available. We have, as we've been hearing about this week, the maternal vaccine, monoclonal antibody, and we also have data showing that both of these have the potential for high efficacy, both against hospitalization, which I'm showing on this slide, and against severe disease amongst those who are hospitalized. But it is important to remember that at this moment we have no head-to-head data. So we don't really know how these two products perform against each other. They look pretty similar in the clinical trials, but it may be that when we see them rolled out, we'll see some genuine differences between them. But in the trials, they had different clinical definitions, they had slightly different settings. And so I don't think we can at this moment really say that one is better or worse than the other. Now, I just want to very briefly think about that mortality question. And so we saw there's about 100,000 RSV deaths a year. I mean, you can see comparison here with some other diseases where there is discussion at the moment. And this really speaks to this issue of prioritization. Typhoid vaccines are being rolled out in many regions, particularly here in South Asia and in parts of sub-Saharan Africa, with a similar number of deaths per year. So we're in that same category as a vaccine that's already been recommended for use in high burden areas with typhoid. But it's far less than the cancer deaths from HPV. So many countries may prioritize HPV introductions. And of course, if you're in Africa, then where malaria is such a big killer, this very much becomes the priority. And there's going to be a huge programmatic effort to roll out malaria vaccines over the next few years. But the question then is, what sort of impact do we think these products could have if they were widely used? Now, clearly we won't get 100% coverage of all babies with either of these products. But if there was 100% coverage, and we assume that severe disease efficacy that's measured is correct, there's about 70% efficacy may also translate into efficacy against death, then that could be as many as 70,000 lives saved. But that so much depends on what sort of coverage is achievable in different settings, and we'll come back to that. And of course, if there was only 50% coverage, then the numbers of lives saved would be considerably smaller, about half that. And as I said, this really is a question for lower middle income countries, which is where the biggest impact on mortality would be. So we do have these interventions already which could have an impact on the burden of RSV. And there are more coming. I missed the slide. Yes, there's more coming, which you can see here. From PATH, this is the snapshot of what's in development at the moment. You can see another monoclonal antibody in phase 3. And then there are several other products, both live attenuated vaccines that we heard a bit about this week already, as well as Moderna's RNA vaccine and a number of other products. So there's going to be more options available in the near future. So how should we think about their implementation? Well, in 2018, PATH put together various objectives of things that we needed to know about, which I wasn't expecting that to happen. There we go. So you can see that critical is burden data, and we find with other introductions, as you were hearing from Danny earlier on, that it's critical for countries to have good information about burden to make decisions about their population, and perhaps more so in the context of disease like RSV, which policymakers in countries, politicians, finance ministries will never have heard of this disease. So the information becomes much more critical than for a disease like malaria, where everyone has heard about it. We also need to have safe products. There has to be efficacy demonstrated, and we need to be sure there's supply. And each of these we need to consider over the next few minutes. And then the systems for delivery become very important. So if we look at these two interventions and consider some of the issues with them, we have data. The global burden data, I think, really is critical. It still won't be enough for all countries, and so more is needed. But let's cover some of these issues. So for the monoclonal antibodies, we've already seen this year major supply constraints both in Europe and in the US. And the reason for this is to do with the current capacity in the system. But it's also more complicated because monoclonals are very difficult to produce at high yield. And so for manufacturing, you have less product for the amount of cost in the production, the size of the manufacturing system. And that does mean that production has a high cost. And so that could mean that even though these interventions may be cost effective, and we've seen some data already on cost effectiveness, the actual cost to countries that have to pay for them may still be too high, and particularly for those Gavi graduating countries that are facing the cost of all of these other interventions at the same time. So monoclonals may be particularly challenging in some settings. Really, innovation is needed here. There are not at this moment new manufacturing technologies that will allow us to have higher yield from manufacturing, and that will, until that is solved, that will keep the price of monoclonals up, and certainly higher than the very small number of cents or dollars that are needed for the Gavi eligible countries. For the vaccine, cost shouldn't be such an issue because we know that with other protein vaccines, these can be produced at extremely low cost. Vaccines can be produced with very high yield. And so in the long run, when the facilities are built, there shouldn't be a problem with supply or the same manufacturing challenges around monoclonals. But we do have the question around safety, which has been raised occasionally during this week so far. And this is from the study from Pfizer, the phase 3 study, which I think I've highlighted here, yes, showed the imbalance in premature births in the clinical trial. And this imbalance was limited to low and middle income countries with about twice the number of premature births with about 20 premature births compared with 11 in the control group, and has caused some concern amongst regulators. And that is highlighted, I think, here when you look at the difference between the approvals that there have been in different countries. So in Europe, because this safety or imbalance of prematurity was limited to low-income settings, in a high-income setting, the European Medicines Agency approved the vaccine for all gestations. whereas the FDA took a conservative approach and licensed from 32 to 36 weeks, even though there was no signal in high-income settings, but have limited the use of the vaccine. And in the UK, the MHRA has gone somewhere in the middle and gone for 28 to 36 weeks. So this already in a high-income setting reduces the opportunities for vaccination against RSV. So we don't really know whether this signal is significant. It could have arisen by chance. It could be it's just circumstantial to that particular trial. But I think importantly, there was also a signal seen in the GSK trial, and they stopped development of their maternal vaccine after seeing it, which suggests that there may have been something going on, at least at that time. So further evaluation of the safety signal is needed, and studies are already planned in some LMICs, as you were hearing earlier from Cheryl Cohen. So could it be possible to avoid this by limiting use to later gestation? And is that actually practical to do that, to reduce the risk of prematurity? Or could countries introduce RSV vaccines, maternal vaccines, with good surveillance to monitor this? So if we look at that clinical trial, the excess prematurity rate was about 2.5 per thousand. So that's the number of additional premature births in the trial. But in low and middle income countries, we know from several studies that the RSV hospitalization rate is between 20 and 28 hospitalizations per thousand. And so perhaps we should be starting to build up a picture of the risk benefit of, even if this safety signal remains, what's the risk benefit analysis of not intervening in this disease, which has a high mortality. Indeed, even in Europe, when you look at from the ReSViNET Consortium, there were 1.6 hospitalizations per thousand, which is similar to the figures there on the prematurity rate of 2.5 per thousand, and also large numbers of medically attended visits. So some risk-benefit analysis, I think, is going to be critical for countries to decide whether to move ahead and introduce the vaccine. I think implementation is also a really important issue to consider. So if we look at the monoclonal antibody, this could be used in LMICs at delivery. And that would seem a good idea, but 28% of global births are at home. They're not in a hospital setting. So it's going to be very difficult to implement the monoclonals at home. And if they're given in the community, they need to be given very early. We heard earlier that BCG vaccine is often given some weeks after birth. But for the monoclonal, you want to give it as close to birth as possible if you're going to have an impact. For the maternal vaccine, the vaccine clearly needs to be given during antenatal care. And the good news is that about 88% of women globally have at least one antenatal visit, according to UNICEF. But is that visit at the right time that makes it appropriate to then give the vaccine? 69% of women do have 4 antenatal visits, and so that gives more opportunity for those women to be vaccinated at the appropriate time. The issue of timing, though, is really important. One of the WHO surveys on tetanus vaccine, which, as everyone knows, is a maternal vaccine, showed that a large number of the doses are given after the baby is born and actually protect subsequent pregnancies, but not the pregnancy that you were trying to protect. So this may be a really important component for RSV vaccines. If they're given to the mother, they might need to be given after the baby is born if it's not possible to give them during pregnancy. And then we need data on how long that protection lasts. Will it last into future pregnancies? And we have no data at the moment to give information about this. It's also important to look at the duration of protection. I think I'm getting an alarm saying my time's up, but I think that's because the clock wasn't reset from the last one. So the duration of protection, the monoclonal and the maternal vaccine, we don't think immunologically that protection falls off a cliff at 6 months. So it may be that there is some protection that continues beyond 6 months. And that makes a difference to many of the choices that might be made for how the vaccine is implemented. But we have very little information about the trajectory of protection from 6 months. There are these questions I mentioned about whether you could give a dose and it would protect in subsequent pregnancies. Should we be giving it in every pregnancy? The studies in the elderly suggested no boosting with the GSK vaccine with a dose given in the following season. So is there any advantage in giving the dose in every pregnancy? And if it isn't given in every pregnancy, it'll be much more cost effective because you won't need so many doses for a population. So we need answers to these questions. So considerable reductions. Now we heard quite a bit about the different approaches that there are to cost effectiveness, and the UNIVAC tool was nicely summarized, so I won't go back into that. For this, both the maternal vaccine and the monoclonal were considered cost saving at a cost of about $3.50 per dose. Some of the other studies which have tried to pull the information together, so this is a systematic review of cost of illness and cost effectiveness studies, show that there is, not surprisingly, a huge variation depending on the setting in which children are born. So in some areas, the cost of illness is much greater than others amongst low middle income countries. And so that does mean that the economic analyses and the decisions in different settings will be very much affected by both the cost of illness and therefore the cost effectiveness analysis. And there are many of the input parameters, we have quite a lot of uncertainty about input parameters over what coverage might be, what the duration of protection is, and so on. And so even though the economic models that we've seen presented give us a framework to work around, it doesn't necessarily tell us the right answer for a particular population. Now, in the title, I was also supposed to speak about seasonality, and clearly seasonality is very important, and particularly if you're in a country like India, where near to the equator, then the RSV season is between 10 and 12 months of the year. Up in the north of India, there's a very clear winter season with RSV. So there's a lot of variation, and so for a country to make decisions about how to implement may be very different depending on the setting. If you have a limited seasonal approach, you'll use fewer doses, it'll be cheaper to implement, and with less product use, it may be that that's the easiest for a country to put in place. However, the impact is much lower with seasonal use, and it's less equitable. There'll be many individuals who might be protected. And also duration of protection may mean that it's better to have a year-round immunization. And indeed, in the UK, when we looked at this, and this is a recent publication with some of the original assumptions in the model, which have been amended for the final decision in the UK. But this model showed a range of different scenarios, from year-round immunization to immunization just within the season, or the monoclonal in the season with a catch-up. And really, when the final analysis was completed, the most cost-effective was to use seasonal monoclonal or maternal vaccine. The seasonal with catch-up for the monoclonal was the next most cost-effective, but year-round monoclonal or maternal vaccine was also cost-effective. So all of those different approaches could be cost-effective in the setting of the UK. But actually, the programmatic questions become much more important for the UK. Switching on the use of a vaccine or monoclonal and switching it off is extremely difficult to do, particularly in a health system which is coping each winter with winter viruses, as well as the flu programs and COVID programs. So it may be that it's much more likely to give you high coverage, more equitable to use year-round immunization. And I think that will be the preferred approach in the UK. So which product and when? Well, the monoclonal antibody, I think, as I've highlighted, implementation remains a challenge in low and middle income countries. And the questions around supply and the cost of the vaccine, of the monoclonal, are going to have to be addressed. And I don't think we're clear at what cost the monoclonal could be sold. What's the minimum cost? Is it as low as $3.50? Or is it somewhat higher than that, which seems more plausible given the production costs of monoclonals? And I'm sure WHO will be doing a supply analysis. But the good news is that there is some competition coming with more monoclonals in the system. For the maternal vaccine, we need multi-dose formulations, and these are expected in the next few years. But there is a risk that these concerns over safety will lead to a delay in reaching populations in the greatest need. And some formal risk assessment is, I think, urgently needed to help inform countries about whether they could implement the maternal vaccine sooner rather than later. The safety studies which are underway will hopefully answer these questions, but they're going to take a couple of years to give an answer. So it's a good news that the WHO SAGE has been formed. You can see on here several people in the audience are members of this working group who are going to be working through some of these issues over the course of this year. And they've only just started. This was established just in December. So there is a really high morbidity and mortality from RSV in low middle income countries. I think it's clear that we need prevention programs in place urgently. Maternal vaccines, I think, are going to be the most accessible for LMIC populations, and particularly if they will provide protection for more than one pregnancy, that could really make a big difference for the cost effectiveness calculations, and importantly for countries looking at budgets for the cost of a program. There are also the questions around access and equity, where year-round may be the optimal approach, but for lower costs, then seasonal may be chosen by some countries. Great progress is clearly already being made, and I think the impact that we've seen this week from the studies in Europe, particularly from Spain, I think really showed just what these interventions could do if they were used around the world. Now, when Louis introduced this meeting, he was very optimistic, but actually, I don't think this is the time to relax. This year, 100,000 children will die from RSV.
Are we going to have the video presentation or the? Okay.
Good morning, everyone. I'm so delighted to be joining you virtually, and my apologies that I could not come in person to be with you today. And I'm extremely delighted to be presenting about how we have to be more mindful of certain aspects relating to equity while addressing this new undertaking of rolling out and scaling up RSV vaccines as a part of maternal immunization in low and middle income countries. As you are already aware, a lot of children die each year due to RSV, and 95% of these deaths happen, unfortunately, in low and middle income countries. We also know that the United States of America, where RSV vaccine has already been rolled out as a part of maternal immunization, it hasn't really been easy for pregnant women to access this vaccine. There have been all kinds of challenges ranging from a lack of availability of the vaccine, the financial impediments because of high out-of-pocket costs, and also wrong use of product. One thing that is important to keep in mind is that the RSV vaccine is entering the immunization space at a time when inequities in immunization are quite glaring and disturbing. 14 million children in 2022 did not receive even a single dose of the most basic DTP vaccine through routine immunization systems, which means they were absolutely zero dose. More than 30 countries have shown coverage trends of either stagnating vaccination or really further declines in vaccination rates. Africa, as a region, remains 5 percentage points below the global level. And if we look at HPV, a vaccine which was introduced 14 years ago, we see that it is still lagging in terms of uptake, with only about 21% of eligible girls having received this vaccine. We've seen in the recent past that new vaccines are becoming harder to introduce in countries, and they're facing a wide range of challenges. So really 2000 was the year when Gavi was set up in order to expand access to new and underused vaccines. And though there has been tremendous success with HIP vaccination, other vaccines have faced a variety of challenges. And you can see here on this graph that HPV still continues to have an abysmally low uptake. We've also seen that new vaccine introductions are not able to address persistent inequities. So zero dose children have remained zero dose and have not benefited from these new vaccines. Increasingly now, we are also seeing more vaccines in the pipeline, and countries are struggling with lack of prioritization frameworks. Fiscal space in countries is shrinking, health systems are strained and stretched, countries are unable to step up investments in health systems, particularly health workforce, and we've also seen misinformation and disinformation growing and really impacting vaccine confidence. Well, if we are talking about RSV, then one important thing to keep in mind is that it is maternal immunization, and antenatal care really is the key platform to deliver RSV vaccines. And when we look at ANC coverage in LMICs, that is quite low in many of the countries where RSV is taking a heavy toll. We are also seeing significant disparities in ANC coverage, and it really is related to a lot of variables like education, age, wealth of the household, and several other factors, really sort of leading to a situation where in low-income countries and also within countries, low-income households continue to see poor access to ANC services. Maternal tetanus vaccine coverage is an important predictor of inequities that will arise once RSV vaccines are rolled out in countries. And as I said earlier, economic status, age, education, residence, whether women are living in urban or rural areas, particularly remote rural areas, they're all important variables or stratifiers of equity. But important to remember that newborn babies of mothers who are not receiving ANC services or maternal tetanus vaccine and therefore face multiple deprivations and exclusion are really at the highest risk of dying of RSV because of their lack of ability to access healthcare in time. That will be faced with regard to RSV by populations are quite similar to those that are faced by zero-dose children. And when we undertook research around the factors responsible for zero-dose children, we realized that 2/3 of these zero-dose children live in households which are subsisting on less than $1.90 per day, which is the international definition of extreme poverty. Also, gender discrimination or gender barriers play a big role, and mothers of zero-dose children are more likely to be uneducated. Ethnicity plays an important role, and here you can see that in Afghanistan there are certain ethnic minorities that are stigmatized and therefore really lack access to essential services represented by a very high proportion of zero-dose children. Also important to remember that nearly half of all zero-dose children live in three key contexts. Urban slums are now increasing, particularly in Asia and Africa, and these urban slums ordinarily lack access to essential health services, including vaccination, and we therefore find a very large number of zero-dose children in urban and peri-urban slums, remote communities where services generally are weak or non-existent, but also nomadic mobile groups which are on the move all the time. And then finally, displaced populations as a result of conflicts, climate-induced extreme weather events, and the number of such populations is increasing, also leading to an increase in the number of zero-dose children. It's very important to remember that there are profound public health implications if children remain zero-dose, because zero-dose children represent households that also typically miss maternal health care. And though 1 in 10 children was zero-dose prior to the pandemic, they account for nearly 50% of all vaccine-preventable deaths. And data shows that these children are more likely to have mothers or who lack access to antenatal care, skilled birth attendants, and also more generally, these households lack access to water, sanitation, nutrition, education, and other services. So therefore, considerations for successful introduction of RSV as a part of maternal immunization and equitable coverage will have to take into account some key considerations. The first, as I said, is prioritization. Countries are having to grapple with competing priorities, and therefore they will require support on understanding the disease burden, the public health consequences of not introducing RSV, and therefore prioritizing this vaccine, then making sure that the health system is strengthened and geared up to actually ensure equitable access, because as I said, populations that are currently marginalized or excluded typically have a disproportionate burden of vaccine-preventable diseases and related morbidity and mortality, and that holds good in the case of RSV as well. Then making sure that costs are affordable, both of vaccine as well as the delivery modalities for the vaccine, for the coverage and progress to be sustainable. And finally, making sure that we invest in understanding community acceptance patterns and the perceptions that community may have of the vaccine, because gone are the days when a vaccine could be taken to communities with an expectation that they would accept it immediately. We have seen the growing challenge of misinformation, disinformation, erosion of community trust in public health institutions and governments, and therefore making sure that we invest in social behavioral research, pay attention to social listening, and actually bring the whole issue of community acceptance and demand center stage will be critical to the success of RSV. And thank you for your attention.
Thank you, Anuradha. I don't think she's online anymore. So we will have the next speaker now for this session, Dr. Marta Tuffet, who's the head of policy at Gavi, and she leads the vaccine investment strategies and the development of evidence-based policy framework to guide scope, allocation, and programming of Gavi funding. She's going to talk about the assessing vaccine for decision-making, Gavi's vaccine investment strategies. Dr.
Thank you very much, and thank you so much for inviting me to this RSV conference. It's been really great to talk with a lot of you, and I'm very conscious that I'm the last speaker on the conference, which was a perfect excuse for me actually to participate in the whole conference. So thanks everyone for engaging with me these past few days. So I'm going to talk a little bit about what Gavi is, as I recognize that not everyone is familiar with the work that Gavi does, and then I'll speak a little bit more about how we prioritize vaccines and within that context, the prioritization for RSV vaccine. So, oops, sorry, very fast here. So I am employed by the Vaccine Alliance, and I'll start off with a little bit of intro on Gavi. So our mission is to save lives by ensuring that we have equitable and sustainable access to vaccines. And since we were established in 2000, we have supported 78 lower income countries, and we currently have 54 countries that are eligible for support. These are the lowest income bracket countries. And through COVID-19, we also supported a wider range of countries. Just a reminder that Gavi in itself is a secretariat, but it is an alliance of different partners. And we have on the board of Gavi a range of different partners, from the multilateral organizations, from the implementing countries, from the donor countries, from civil society, from academia, and the manufacturers. So it is a wide range of different stakeholders, and together we have the opportunity to make a big difference with vaccination across the world. And in terms of the model is really based on the fact that vaccine market forces alone were not meeting the needs of those that needed vaccines the most. And so by being able to pool the demand of the lowest income countries, we are able to provide some certainty for manufacturers and incentivize manufacturers to come into this market and ensure that there are products that are available and affordable for those that need them most. So we are procuring vaccines for half the world's children, so it's not an insignificant market, and that's really what has driven the success and has been able to drive prices down. So if you look at a Gavi country, an EPI immunization schedule would cost something like $28. By comparison, in the US, that would be about $1,000. So we've really been able to drive market prices down and create a healthy market for manufacturers to compete and to provide vaccines for a share of the market. We also have an approach to strengthen vaccine delivery platforms. So we do provide some health system support, and we also have a sustainability model. So Gavi does not support countries in perpetuity. We support according to the ability to pay. So as a country's GNI per capita increases, so will their contributions to the procurement of the vaccine. So it's a co-financing model. We provide only a proportion of the total cost of vaccines for a country's birth cohort. So these are the sort of three types of support that Gavi provides. In the middle, it's the primary type of support. It's the vaccine procurement support. But I mentioned we also provide some cash support to countries through our health system support, and we also provide technical assistance through our partners like WHO, like UNICEF, like World Bank, like civil society organizations, etc. So let me talk about how Gavi makes decisions on how we prioritize vaccines. And we've heard a lot about the challenges in terms of prioritization in light of a lot of competing priorities in light of fiscal constraints. And we also have to think about that in the context of Gavi. What we do is we take a look at what vaccines might become available in the future and do an analysis to help inform that process. So you can see here, over the course of Gavi's life cycle, since it was established in the year 2000, we have added new vaccine programs to the portfolio at every single cycle, at every single 5-year cycle. So we now provide vaccines against 19 infectious diseases through 46 product presentations. So we have multiple product presentations per infectious disease. We're currently in the 5.1 strategic cycle that runs until 2025. And we are now assessing the potential vaccines to include in the next strategic cycle from 6.0. What's important to note here is that back in 2018, when we were assessing what vaccines to add into the current cycle that we're in, we did assess the RSV vaccine and the monoclonals. And a decision was taken by our board then, an in-principle decision, that as and when those products became available and contingent on meeting certain criteria, that they were licensed, that there was a pre-qualification, and that there was a SAGE policy recommendation and the cost assumptions were met, that these products would be introduced. Unfortunately, the product that was in the lead did not meet its clinical endpoint, so we have been following since then the pipeline, bearing in mind that decision in principle. That decision in principle still stands, and we're following the subsequent products, and which is why we're very interested in discussing with all of you to understand what are some of the implications. So let me talk a little bit now about the vaccine investment strategy itself and how we make the decisions, and what are the criteria that we use. So it is an evidence-based approach that we conduct every 5 years. And it involves a transparent methodology. There's significant analytical review of existing evidence, but we also commission modeling, and we have extensive consultations, and we bring in independent expert advice to inform that process. It helps our strategic decision making. So rather than having a first come, first served approach for when a vaccine is available, we're able to take a more holistic view and then see what are the trade-offs and what's the best value for money. Importantly, it provides advance signals to vaccine developers and suppliers. It gives a flag to say, hey, there might be a potential market, Gavi market here, and they are able then to make their own risk-based decisions on whether to continue to invest or to strengthen some of their investments. But likewise, it also provides predictability for donors because it informs our resource mobilization strategy, and it provides also predictability for governments who can also start thinking about their potential prioritization processes. And the types of investments that can result from it can be vaccine programs to introduce into routine, or global stockpiles or learning agendas, global stockpiles in terms of outbreak response. So it's an analytical process. It has 3 board decisions, and it's aligned with strategic development. And throughout this process, we tend to ask these different questions. What are the vaccines that we will evaluate? How will we evaluate them? What's the burden of disease? What is the appropriate vaccination strategy? What will be the demand? Will it be feasible to implement? What would be the potential impact? How does it compare to other potential available products? Or how does it compare to the existing portfolio? And the further a vaccine product is away from development, the harder that some of these questions are to answer, and we have to make a lot of assumptions, and we bring groups of experts to help us make those assumptions. So in terms of how do we evaluate them, we have a number of criteria that we use, which are ranging from health impact, value for money, equity, social protection, what are comparative advantages from a market shaping perspective, what the economic impact, indirect or direct, and then we have a series of modulating criteria that help us modulate up rather than down. So for example, under global health security, we will be considering impact from climate change or impact on antimicrobial resistance. And implementation feasibility, we'll be considering things such as how feasible is it to introduce this vaccine into a vaccination schedule? If it's outside the EPI schedule, that poses additional challenges. What is the cold chain storage implications? The bigger the packaging, the more space it takes up in a fridge. So we take into account all of these different types of considerations. Is it difficult to administer? So will a health worker need to be retrained to be able to deliver this particular product? So these are all factors, and we have qualitative and quantitative indicators that sit underneath these criteria that help us inform our analysis. I'll skip this one. I'll skip this one as well. So currently, we have an existing portfolio that is primarily within the EPI schedule, and we have ventured outside with HPV in the adolescent stage and with a few vaccines in the second year of life. In 2018, some of these vaccines, including RSV, spread us further along into the life course, and currently we are assessing a number of different new vaccines that are in the pipeline, such as a maternal group B streptococcus vaccine, a Shigella vaccine, a dengue vaccine, a new TB vaccine for adolescents, and whether to continue or not to continue with COVID-19 in the future, as well as a hepatitis E stockpile vaccine. We're in the process of doing that analysis at the moment, and here's some initial data in terms of the comparative analysis of the impact of these vaccines in terms of the deaths averted versus the current Gavi portfolio. So in green is the current Gavi portfolio, and in blue is the new portfolio. So just to give you an idea that we basically try and take into consideration the full portfolio. So with RSV, where do we stand? So as I mentioned before, we do have the decision in principle. So we are a bit further ahead than with some of the other vaccines that we're considering for the next strategic cycle. And there was an assessment that was carried out already in 2018, as I mentioned, but unfortunately the product that was in the lead did not meet its endpoint. So at the time, in order to make the decision on whether to include RSV into the portfolio or not, we carried out some analysis. We had some initial demand and price forecasting, and I'll show you a little in a second what that looks like. It's out of date, and so please don't take too much notice. We will be updating that, but it's just to give you an idea of the type of data that goes into our decision making. And we had significant amounts of consultations with countries, with partners, with CSOs, manufacturers, disease experts to help inform that. We had engaged with PATH, who are here on some of the modeling, as well as with London School and with the Gates Foundation on that. And this is the type of output that we get. Again, don't take too much notice in the numbers. This is out of date. This was carried out in 2018, and it was based on assumptions at the time, which might not necessarily hold today. But you can see there's a real wide uncertainty range as well, which is based on assumptions that we made about the potential variable efficacy, the duration of protection, and the coverage that we would hope to achieve. And so this is the type of information that we would generate for each of the vaccines to help inform our decisions. And here you can see the same for the number of cases as well. And we will be updating these analysis over the course of this year. And so this is how we ended up with RSV in the Gavi portfolio decision in principle. So at the time, we were looking at other vaccines like rabies, DTP boosters, Hep B birth dose, as well as maternal influenza, dengue, and Hep A. And this is how RSV scored in terms of the different criteria that I mentioned before. You can see that it was on the lower range of some of the other vaccines that we were considering. However, it did make it through. Dengue at the time didn't make it through. However, we have reassessed this as part of the new vaccine investment strategy, and dengue is currently in the shortlist, although a decision has not been reached. So in terms of where we're next in our assessment timeline, so we have the decision that was made in principle in 2018. There was some discussion about whether RSV should be reevaluated with the current new vaccines that we're considering. And the conclusion was no, that we still have a decision in principle with RSV. We should continue to hold true to that, but we need to update our analysis to help inform that as well. So we would need to go back to our board, but we're in a different stage that we are with some of the other vaccines. I mentioned that there's a number of different coordination on a number of steps that we need to reach. And the first is that we're expecting a SAGE recommendation for the maternal vaccine at the end of this year, which is one of the criteria that we need to meet in order to open a vaccination program. So in terms of where we are, we will be carrying out updated demand and price forecasting. We will be trying to assess some of the health system strengthening needs to introduce and deliver a product successfully. How do you integrate it into the ANC services, for example? What will be the acceptability? And then we will also be calculating what are the financial implications for Gavi and for countries, and what are some of the programmatic considerations and some of the pre-introduction activities that we could think about, and then how do we align it to the new Gavi strategy that is currently in development. And so rather than getting you to ask me questions, I thought I would ask you some questions. So these are some of the questions that we have, which I'd like to pose to all of you in terms of where our thinking, the data needs that we need to help inform our next steps with RSV. Thank you very much.
Thank you very much. And the house is now open for any questions or comments. Anybody? So, Dr. Naveen, you have some? Okay, go ahead, please.
There's been some mentions of revaccination and subsequent pregnancies. And I was wondering if anyone could comment on what assumptions are being considered for protection that may be offered in subsequent pregnancies with or without dosing, given lack of data among pregnant people and some limited data among older adults. Thanks.
I think at the moment the duration of protection or the possibility of protection in subsequent pregnancies has not been included in the models, but maybe the modelers here should answer that question rather than me. But as far as I know, it's not included because we don't have any data.
Yes.
Hi, thanks for the great talks. Martha, I have a question for you on Gavi. The number of countries that fall under Gavi is, as you mentioned, there's a number of countries that are transitioning out, and they're then required to put in more of their own resources. And as the number of vaccines increases but the burden may still remain, particularly in these low, middle to middle income countries. What are your thoughts there? Because the burden still exists in many of these countries, but the burden of cost will then be borne more by the countries as well.
Thanks. No, that's an excellent question, and it's a question we get asked on a number of different diseases, including some of the vaccines that are in the pipeline. A dengue vaccine, a chikungunya vaccine, the TB vaccine. The burden is primarily in the middle income country categories. Gavi does have a middle income country approach for a subset of vaccines that are considered priority. For example, PCV, Rota, HPV. However, we do not provide a significant amount of funds for that. It's mainly around being part of the the support family, the technical assistance that countries can get, and some accessibility to some of the pricing. For RSV, indeed, there is burden beyond the Gavi countries. However, I think it is part of the success of the Gavi model has been the sustainability co-financing model, where countries do take increasingly more ownership of their procurement responsibilities for vaccines, and that has really led to some strengthening of the health systems in countries to be able to have a sustainable vaccination strategy. And so since Gavi was set up, we've been able to mobilize more than $1.5 billion in domestic financing for vaccines, which I think is a huge success. So I think there are pros and cons, but one has to consider also the sustainability element as well.
Yeah. One comment. So I served on Gavi board for 6 years, but still VIS always, I really never understood fully. We do it every 5 years. So what if there is a, in between there is no chance, and what's like you said, some are deprioritized, then we consider as antigen by antigen, but not even their, like combination, like we will give MR, but not MMR. Because mumps is considered separately and it's not considered a public health problem. And so we in India are now seeing a lot of mumps cases, plenty. Second, previously there was, we give penta, but we don't give booster. So is there any change in approach now, or it's still same? And what happens before 5 years? And why don't we consider this as a, in one piece? Like if there is a MMR vaccine, why we just exclude and promote MR? And some, how we started giving booster doses because you want to prevent, say, disease from penta, but then you don't want to prevent after 1 year. So you are just delaying it. Is there any change in approach or not?
Thank you. So the 5-year cycle, it's to inform the next strategic period. And if you go much earlier than that, then the probability of technological success of a product is less certain. So it's harder to make assumptions about the potential impact. If you go shorter, you're not given sufficient advance warning to the manufacturers to be able to plan ahead as well in terms of their investment. So it is a fine balance. We do have the opportunity to make real time decisions in the context of a public health threat. So we have the opportunity, and we do carry out living assessments of certain vaccine or certain diseases according to WHO's R&D blueprint list of prioritized vaccines considered of epidemic and pandemic potential. So we do have that mechanism. We are thinking now about, that age range as well with the advent of mRNA vaccines, and, is that the right time? On the question about combination, so yes, indeed, it is something that we're considering increasingly, and I think there's generally a need to have a wider discussion from the global health community about what are the right combinations. Manufacturers are coming to us. What is the right combination? What do countries want? The EPI time point is really busy. How do we address that? On boosters, we have actually now introduced DTP boosters for older age groups, and so this is completely new, almost hot off the press. So countries are now able to apply for DTP boosters as of this year. So there is some progress in that, and we're really excited about that. Thank you.
Yeah, please.
So I can ask you a question as well. One of the big issues for country implementation is actually advocacy. And even with the extraordinary amount of advocacy that went in around pneumococcal vaccines, we're still not seeing the uptake where it should be in low and middle income countries. And so I wondered when in 2018, when you said you had engagement with governments in different countries, had they ever heard of RSV? And is there actually a major investment needed just in raising awareness?
Yes, we do. we do engage with countries, and actually at the start of the current VIS process, we went back to countries, and there isn't a huge amount of awareness at the country level in RSV, and we think that that's one of the reasons why it's scored low. I don't know if you remember, Danny, if I can presented a slide on how countries, EPI managers mainly, were ranking different vaccines, and RSV was right at the bottom. So there is generally going to be a need for a lot of demand generation. But when we did the survey in 2018, what was really promising is that the feeling was that there would be a high acceptability. So there is that. Of course, when we're serving these countries, EPI managers, they're faced with current outbreaks, with current challenges in routine immunization. It's very difficult to often project yourself to 5-10 years from now. But we don't have the benefit that we have with malaria or with TB, that are very obvious diseases that the community is very familiar with.
Yeah, go ahead, please. Thank you so much for this great ending session of our meeting. And my question is for Andy. So Andy, you pioneered one of the first COVID vaccines in record time. And I just wonder, how do you see new vaccines that come to the market? What would be the time lag from development? What would be the time lag for licensure? Because of course COVID was a pandemic, we were in an emergency situation, but how can these lessons learned from COVID we can take for new vaccines? And for example, in your institute, the new product development, is it really streamlined? How new products will come out after the lessons learned from COVID? Thank you.
It's a great question. I mean, a lot of the challenges with vaccine development are not the scientific ones, where there's obviously many products, and here we're looking with RSV. The challenges ahead are not really the science of making vaccines. We've got vaccines and monoclonals, a number of those products. A lot of it is what happens after that. It's the processes of showing governments that there's a burden of disease, working out the financing, working out the program implementation of them. And that's true in high-income countries as well as low and middle-income countries. But with weaker health systems, it's even more challenging. So I think the actual development process, the speeding that up, there were obviously big regulatory advantages in the pandemic with rolling submissions and so on, which shortened those timelines. But I think one of the other key differences was that rather than the long periods between phases of clinical trials, which happens in peacetime, the phases just ran straight into each other, because there was a financial risk taken, usually by governments funding those programs, that meant that the trials could just keep going. Whereas normally you wait for often a year or more between phases, looking at the results and persuading, if you're an industry, senior management from the next stage, or if you're in academia, finding a funder for the next stage of development. So I think a lot of the delays normally that are built in are back again because we're back in peacetime, which is around the financing of development as much as it is about the other processes. The regulatory process certainly in some countries, I think, are better than they were before the pandemic, but that's not true everywhere.
More questions or comments? If not, Dr. Naveen, you want? No, you want to give your final comments?
No, I think it was good, very good presentations, and we have finished before time. Thank you so much.
Yeah. Good afternoon to everyone. So please don't leave the room because we are getting close to one of the most exciting moments of the conference. And my co-chair, Ashley Mara, will join me in disclosing who is the winner and which is the ranking of the Young Researchers Tournament. Please, Ashley, welcome. Can I have the slides, please? It's already here. Wonderful. So, as you know, ReSViNET has been committed to organize this Young Researchers Tournament to encourage young scientists to hear their ideas and to give you and to emphasize their commitment and their enthusiasm. And we rely very much on young people to take the relay and go on with the research and with the mission of ReSViNET. So they are very welcome, and they have always been. This year we had 51 candidates from 15 countries, and they were selected after a very careful assessment and evaluation. And we went out, as you know, last day with introducing 4 oral presentations by 4 of them. This selection was based on the merits of these abstracts, including innovation, quality of the research, clarity, and also each individual performance on the scene, on the stage, addressing the questions and expanding the engagement with the audience. So we are ready to announce the ranking. As you know, you were invited to vote for a while. There has been a very careful and deep assessment of votes and eligibility of votes. Now we have the final results, and I will have the honor to announce the fourth, the third, and the second. And then Dr. Ashley Murray, who is the winner in charge, will have the pleasure and the honor to announce whom will replace her for this year. Okay. Yeah.
So these do come with a cash prize for the winners, and we'd like to highlight that this is to go towards your continued scientific endeavors and your professional career development. And if the winners could stay off to the side for pictures at the end.
Not only pictures, but also the certificate award. And actually, one thing that we were told to remember, this money comes from the ReSViNET Foundation. And it's a pleasure to award this this amount to these winners. So we are ready. Fourth place, Tangan Yannick. Tangan, are you here? Oh, I see him. Please join. Thank you, Tangan. The final next steps towards success. Well, thank you, Tangan. So now we go into the third place, and the third is Mary McCabe. Yeah. Mary McCabe. Tangan was from Cameroon, University of Cameroon. Mary from Belfast. The interferon scientist with us. So almost ready. Second place, that obviously will also imply that we know the first one. Yes, the second is Anubama Rajan.
I guess it's me now. So first place is going to Jonah Verstappen. Mhm.
She obviously now is in charge, so she needs to close this award ceremony.
Wow. Okay. Well, all I can say really is thank you so much for having all of us, and congratulations to the other participants as well. It's been an honor to be here. Yeah. Thank you.
Well, thank you, Ashley. So we are very happy to leave the microphone and the stage now to the masters of ceremony and chairs and driving engines of this conference, Louis and Leila. Thank you very much.
So thank you for all for staying until the end. It's my pleasure to close off this RSVVW 2024 together with my partner in crime, Leila. Of course, I'm very happy that Jonne from Team Netherlands was there. I voted 27 times, I have to confess. I want to thank all the people who organized this meeting. I think you have seen that a lot of effort has been put into getting where we are now, coming here to India, in Mumbai. I've heard a lot of positive feedback from people that we should keep on coming to countries like India. Thank you. That means a lot to us and to the rest of the team. I'm going to switch to the next slide. With 422 participants, many sponsored symposia, so many scientific sessions, master classes. Also, Keith, thank you for jumping in for Barney, the Young Researchers Tournament, and 191 abstracts. I think it has been really a big success. There are two things that stand out for me before I give the microphone also to Leila. One is the feedback that I heard about the flow of the conference, that there was enough room for interaction and going to watch the posters, and we are definitely going to keep it organized in the way that we have plenary sessions and that there is enough time to really enjoy a meeting together with the RSV folks around. The other thing is more at the scientific or impact level. Danny, next time we make sure that RSV is higher at the Gavi bar than just at the border of being funded. We will make sure that it is getting its fair piece of the global investment. Leila, I think you're going to take over for the final words.
So, sorry. So looking back, we once again wanted to thank you to our sponsors, and also to that we had so many company-sponsored pre-symposiums, which was and also the great people that attended those symposiums. So that was also new to our conference, and that was a good sign. Just a few pictures we want to just bring in, just to show you how everything went, and also how great it was. We had, well, the room was actually full almost the whole time. Even now at the last moment of the day, it's still being open, full. We had a lot of discussions indeed, as you said, Louis, and I think one of the highlights was our inspirational speaker, Dr. Pavan Agrawal, and about his Dabavala team with 5,000 people here in Mumbai. I think it was very inspirational, and we received a lot of nice and really, I think it was Rhonda Gunn who said this is one of the in top 5 best presentations that I ever had before. So that was, I think, one of the biggest highlights, I would say. We had good, the poster sessions were attended very well. So thank everybody for bringing in your posters here, and also for the participants who attended the posters. A lot of good discussions. We had, of course, the Barney Graham Award for Heather, and hopefully in 2 years we will have, of course, a new Barney Graham Award. So if you know anyone that you want to bring forward, you can always come to Louis and me. The master classes, so many thanks, Rick Robey and also Keith Klugman, for providing the master classes for our young researchers. Okay, so just some pictures for you to see how nice it was, and also the dinner that we had yesterday. So did you miss a session, or did you, also for people who are not able to be here, we have all the conference is has been live streamed and also being recorded, and it will be uploaded on the ReSViNET website. Just coming back here, also the presentations, we have asked speakers to provide us their acceptance to share the slides with you. So within, let's say, a week or so, the presentations will be added to the website. Then I want to thank to our organizing committee and chairs, so Heather and Joyce. Please join us again. It was you have been with us from the beginning. I'm not sure if Heather's still here. Oh, come with us, Heather, if you want. Just, I think so you deserve to also stand here. big walk, right? From
While Heather, just take your time, is here, I just wanted to point out that the entire scientific program was directed by our chairs. We had a lot of support from the scientific advisory group, but if you liked the science, it's all in the hands of Joyce, Barney, and Heather. Thank you so much, and please give them a big hand. And this, I'm sure this is a good moment also to call the rest of the team, Leila.
So we would have not been here, and we would have not have this great conference, the organizational part, and also the good, I don't know, the good food and everything, the music and everything that you have seen, if it wasn't for a few people from my team. So I would like to ask Sophie, Roxy, and Marion to come here. So I must admit that my team was not happy when we said we are going to India and they had only 10 months to arrange this conference. So there has been a lot of, I don't know, it's a Dutch saying, there was a lot of sweat, blood, and tears. So let's say there was no blood, but tears but I think it was worth it. And so we don't have flowers for you because you're all going to travel tomorrow. So we are not going to do that, but we will provide with the team. We will go absolutely on a dinner together, and we will have a good day. And we are all back in the country. I do want you to give a big applause to the team once again, because they really and also just big thanks to the rest of my team, because if they weren't able to cover the rest, then it wasn't possible for these ladies to do the conference part. So also to Liz and Louisa, many thanks for being a great assistance. So I think the last part is one of the last slides is your feedback is valuable. So we really appreciate your feedback. Any feedback that you have on the quality, the place, the venue, the science, maybe how we can involve different countries or different age groups, or I don't know. So every suggestion is valuable. So we have this QR code, and I believe it will also be sent to you by email. So don't worry if you don't have the scanner with you. If you fill out the survey for your feedback, you can also earn 22 CME credits for the conference. So we want also something from you. And at the end, just saying that we have, once again, thanks to our sponsors. It wasn't possible without you, so thank you for supporting us. So as you have heard before, so next year the conference will be organized by ISERV, the RSV Society, and that will be in Brazil. So we hope to see you all there. But the next ReSViNET Conference will be in 2026, and we are going to Europe, and we are going to the Mediterranean region, but we will keep it, we will keep it for us where we are going, so you will hear it in the coming time. Stay in touch. Once again, thank you on behalf of ReSViNET. And that's it.
Thank you.
Thank you.
Safe travel.
Thank you then. Hey.
Stay for a drink.
Oh yeah, there are some drinks if you want.